So my name is Bruce Walker. Good morning, everybody, and welcome to the Ragon Institute, which we're always delighted to show off, and also to the Massachusetts Consortium on Pathogen Readiness annual symposium, which is really a gathering that brings together scientists, clinicians, industry partners, and many others committed to advancing research in infectious disease and immunity. Over the past several years, I think our community has really been challenged, not just scientifically challenged, but also challenged in terms of how we can even work together. And out of those challenges arose something really special, the MassCPR, which is a collaboration of faculty from Harvard, MIT, BU, Tufts, and UMass, as well as the hospitals and industry and the Department of Health, as well as dozens of other institutions. So from addressing emerging pathogens to studying long-term consequences of viral infections, we have about 2,000 people that are members now across the country. And they've shown, I think, really convincingly that the more interconnected we become, the more that becomes possible for us as a scientific community. The MassCPR has shown that aligning efforts across disciplines and across sectors allows us to move faster. There's no question about that, to think more creatively and to achieve outcomes that once really seemed out of reach. And I have to say personally, in my 45 years in this community, I've never seen something quite so transformative. I know hundreds of additional people that I never would have known if it hadn't been for the MassCPR. I think it's really brought us together as a community. The symposium really celebrates that spirit of collaboration and explores potential at the interface of academia and the pharmaceutical and biotechnology industries. Academia drives curiosity, discovery, and a deep understanding of mechanisms that underlie disease and immunity. Industry brings the ability to translate that knowledge, to translate it into products that reach patients like vaccines and diagnostics and therapeutics, and that change the course of lives, and in cases that we will all experience, changes history itself. Collaboration isn't just about handing off discoveries. It's about co-creating science, and that's what really we're trying to do today. Designing studies together, sharing data and tools, asking the right questions from the start. It's about building trust and understanding about each other's perspectives. When academia's fundamental science advances meet industry's focus on deliverables, innovation accelerates. We see this in action today. Basic discoveries in viral pathogenesis are paired with novel platforms in drug and vaccine development. Advances in immunology, systems biology, and artificial intelligence are helping guide smarter trials and helping to improve understanding of host pathogen interactions. I think it's correct to say that we're really working together to make the world a better place. Today's speakers will explore these themes from multiple angles, from the bench to the bedside and from discovery through implementation. You'll hear from keynote speakers bridging basic and translational science, shaping how we prevent infectious diseases. You'll also hear from trainees poised to deepen the connection between academia and industry. Both see the potential that lies ahead, and we need to be the catalysts that really make that happen. As we begin today's program, I encourage you all to really engage with one another. Ask questions, challenge assumptions, and think boldly about what's possible when we work together. What unites all of us is a shared goal to improve human health through rigorous, creative, and impactful science. No single institution, discipline, or sector can meet these challenges alone. Progress depends on partnerships and on sharing ideas, data, and responsibility. So thank you to all of our speakers today, all of the participants, to the organizing team, and to the funders who made this gathering possible. I look forward to a day of thoughtful discussion, new connections, and exchange of ideas that will hopefully continue to drive progress in infectious disease and immunology research. I'll hand it over now to Gaurav Gaya, an associate professor at Harvard Medical School and a core member of the Ragon Institute, who's going to be the symposium moderator today. So Gaurav.
Thank you, Bruce, for that wonderful welcome, and excited to have everyone join us for the symposium today. I'm going to start by introducing our first keynote speaker, Pirada Allen. So Pirada is the research head of seasonal vaccines at Pfizer, where she leads the team developing RNA-based vaccine platforms for viral targets and oversees the company's influenza and COVID programs. She brings 15 years of pharmaceutical experience, primarily in influenza vaccine research and development at Novartis, and also at CSL Seqirus, and she's worked across multiple platforms to improve seasonal vaccines and strengthen pandemic preparedness. She earned her PhD here in biological sciences and public health from the Harvard School of Public Health, and we're really welcome to bring her to the stage.
All right. Just checking my microphone. Okay, cool. All right, so good afternoon, everyone, and thank you, Gaurav, for the introduction. And before I begin, I also want to thank the organizers of MassCPR for giving me the opportunity to speak today on behalf of Pfizer. So I've titled my talk From Moonshot to Flu Shot. So moonshot was a word that Albert Bourla, so Pfizer's CEO, had used to describe the development of the COVID-19 vaccine. And in full transparency, I wasn't working at Pfizer yet when the COVID vaccine was developed, but the successful moonshot inspired me to join the company about a year later. So today I'll be talking about how some of the breakthroughs and insights from the COVID-19 vaccine development can be applied to influenza to hopefully move us from a moonshot approach to a more robust flu shot strategy to combat potential future outbreaks. So this is my conflict of interest. I'm an employee of Pfizer. So as this audience well knows, there are many essential components required for pandemic readiness. And so just at high level, we need global surveillance and early detection systems to quickly identify those emerging health threats, including the monitoring of zoonotic diseases and the rapid sharing of genomic data. We also need vaccine platform readiness for accelerating vaccine development and deployment, ideally within a context of a very flexible regulatory framework that can be adapted as the situation evolves. Then aside from vaccines, we also need point-of-care diagnostics and therapeutics, and these also need to be scalable and rapidly deployable. And of course, global coordination is important, especially alignment between the WHO and regional networks to ensure a unified response. And of course, with clear and open communication to maintain public trust and for the accurate dissemination of information. While all of these are incredibly important, today I'll mainly be focusing on vaccine platform readiness. So this slide just highlights some of the essential features of a sustainable vaccine platform for pandemic readiness. So first, you need to have rapid development and scalable production capabilities. And that includes the quick design and synthesis of the vaccine, a streamlined manufacturing process, and a robust global supply chain that can be supported by warm-based manufacturing. And then second, adaptability is key. So this platform needs to be able to quickly respond to emerging pathogens, ideally using a plug-and-play approach to incorporate new antigens, and can also accommodate a multivalent vaccine if needed. The vaccine should also be stable. So a lot of the stability improvement work that Pfizer did on the COVID-19 vaccine was focused on thermostability to reduce the dependence on the logistics of ultra-cold chain requirements for the vaccine, and also to expand shelf life. And finally, to ensure global vaccine access, we need efficient logistics and infrastructure for rapid distribution, alongside low-cost manufacturing and other preparedness tactics to really ensure affordability and also predictability of access during a pandemic situation. So with all of these considerations in mind, among the different platform technologies that are available, mRNA vaccines emerges as one that is ideally suitable for responding to a pandemic, given the agility, the flexibility and versatility of the technology for rapid development and quick production scaling. From a biosafety perspective, it's also ideal. You don't need live virus, so all the components are non-infectious, chemically defined. And now we also have a huge safety database based on our experience with the COVID-19 vaccine. And the platform has also demonstrated efficacy, eliciting a very multifaceted immune response, eliciting both antibodies and T cells that can contribute to effective protection. What some people might not realize is that Pfizer had demonstrated an early commitment to this technology even prior to the pandemic. So in 2018, Pfizer had secured a license agreement with BioNTech to develop an mRNA-based vaccine for seasonal influenza. And I think it was this early adoption of the technology that really allowed the company to quickly pivot to SARS-CoV-2 when the COVID-19 hit in 2020. So aside from the early development, there has been a number of key milestones for both mRNA and LNP development that occurred prior to the pandemic. But here I'm highlighting just three what I think are scientific breakthroughs that really laid the foundation for the rapid development of an effective COVID-19 vaccine. So first was discovery of modified mRNA. So work that was done by Katie Carrico and others demonstrated chemical modification of the mRNA, particularly incorporation of the modified nucleosides, can reduce its innate immune activation, enhance RNA translatability, and also increase antigen expression, making mRNAs safer and more effective for use as vaccines. And then second, work done by McClellan's lab on identifying the two key protein substitutions in the spike antigen sequence of beta coronaviruses to keep the spike protein in a stabilized pre-fusion conformation was absolutely critical in an understanding of how best to design the SARS-CoV-2 protein to elicit a strong and protective immune response. And then third is the creation of delivery vehicles, the lipid nanoparticles in particular, that enabled the safe and effective transport of mRNA into target cells. So these three discoveries really paved the way for Pfizer and BioNTech to rapidly develop and deploy the mRNA vaccine during the pandemic. So as I think this audience probably very well knows, the COVID-19 vaccine development was truly unprecedented in terms of speed, scale, and global collaboration. And so just to highlight a little bit of the timeline, it was the 11th of January 2020 when the genetic sequence of SARS-CoV-2 was first published. And then a little over 2 months later, the very first COVID-19 vaccine candidate entered human clinical testing. And this really rapid progression from sequence to the clinic was unlike anything we've ever seen before. And then by around the May timeframe, there was over 150 COVID vaccine candidates in development, spanning a number of different platform technologies, as you can see here. And this diversity of approach truly reflected the urgency and innovation that was driving the pandemic response. We also see collaborative efforts across public, private, and academic sectors across the world, and this collective effort was truly essential in accelerating vaccine development and deployment. And then ultimately, less than a year after the SARS-CoV-2 sequence was first published, on the 11th of December 2020, the very first COVID vaccine from Pfizer and BioNTech received emergency use authorization. And so overall, this COVID-19 vaccine development experience, I think, really set new standards for what was possible during a pandemic and highlighting the power of scientific innovation, regulatory agility, and also global collaboration. So this slide just highlights the global impact of Pfizer and BioNTech's COVID-19 vaccine community. Since it was launched in 2020, I think now over 5 billion doses have been shipped, ultimately reaching 183 countries and territories worldwide. And so this global scale reflects not just the reach of mRNA technology, but it also showcased Pfizer's in-house end-to-end vaccine capabilities, from the rapid vaccine research and development to the scalable supply and manufacturing, to the distribution of safe and effective vaccines on a global scale. And it also highlighted the power of global collaboration, working with governments and organizations and health authorities to ensure access and impact. So this, I think, showcases the potential of leveraging the mRNA vaccine technology to combat not just COVID-19, but also potentially future pandemics as well. So we all recognize that SARS-CoV-2 has now transitioned from a pandemic to an endemic state. However, there's still ongoing need to provide COVID variant-adapted vaccines to ensure continued protection, especially in the most vulnerable population, the older adults, the immunocompromised, and those with underlying health conditions. So overall, one of the key lessons learned from the COVID vaccine response was the ability to adapt the vaccine very quickly to emerging threats and under extremely tight timelines. So this COVID-19 working model shares some resemblance to the seasonal influenza strain change process, although it's much less well established and under an even more condensed timeline. So at high level, this process starts with ongoing surveillance and preclinical development activities that are conducted at risk to really identify and assess the different potential variants that are emerging. And then once a variant is selected, and this is a decision made by the WHO and the FDA through their VRBPAC committee, manufacturing can ramp up very quickly. And this process between strain identification to vaccine rollout is less than 100 days, including all the regulatory submission and review process. Now, with the current new COVID vaccine regulatory framework, manufacturers now also have to conduct some additional post-authorization studies, including looking at the safety and immunogenicity of the updated vaccines in target populations. But really, this rapid and coordinated approach prior to this vaccine rollout, I think, represents an advancement to traditional vaccine timelines. And this COVID-19 working model could also serve essentially as warm-based manufacturing for potential future outbreaks. So as I described, really evaluating and updating this COVID-19 vaccine is really a multifaceted and continual process. So for variant surveillance, Pfizer leverages both external and internal proprietary algorithms and modeling to generate a very integrated analysis of genomic, structural, and immunologic data to track the emerging variants, to assess their immune escape potential, and also to anticipate which variant will become the most dominant and pose the greatest risk. We then conduct preclinical studies to assess variant escape and also to better understand the immunogenicity of these new vaccine formulas. So at Pfizer, we typically leverage mouse immunogenicity study models with different immune backgrounds. So we conduct studies both in animals that are vaccine naive to better understand the relative antigenic distance of the different variants. And this model is also particularly relevant to the youngest pediatric population who may not have been exposed to infections or vaccinations before. But we also conduct the studies using vaccine-experienced mice to better approximate the mixed immune background of the general population. We then conduct clinical studies to confirm the favorable safety profile and to confirm the immunogenicity that we observe from the preclinical studies. And this, of course, is supported by real-world evidence to monitor the vaccine effectiveness and the impact of changing variants in a diverse population. And this real-world data really guides our decision for the following season. And in the middle of it all is safety. So we conduct continuous safety monitoring and risk management throughout the life cycle process to ensure that these updated vaccines maintain a favorable risk-benefit profile. So we share all of the data generated here with advisory bodies, such as the WHO TAG COVAC, to help inform decisions, which ultimately is made by the WHO and FDA and EMA, on whether a new variant is needed for the following season. And I think the key strength of this approach is that the trends that we see in preclinical models actually is quite consistent with the trends that we observe in our clinical studies and our real-world effectiveness data. And so this alignment, while not perfect, gives us some sort of confidence that what we observe in the mice will be somewhat predictive and consistent with what we'll see in humans and in the real-world population. So just to take last year's variant-adapted vaccine as an example, so here on the leftmost side, the XBB 1.5 vaccine was shown to induce a very strong neutralizing antibodies response against the XBB 1.5 virus, as shown in the blue bars. However, when we tested it against a newer JN1 virus, we saw about a 7-fold decrease in neutralization titers. And so this trend was also observed in our clinical studies, which is in the middle panel, although with a smaller 2-fold decline. But this reduction in neutralization titers also corresponded to reduced vaccine effectiveness. So on the right, we're looking at the XBB 1.5 vaccine effectiveness against hospitalizations, and you can see that it drops from 62% during an XBB dominant period to 32% during a JN1 dominant period. So this prompted us to evaluate an updated COVID variant adaptive vaccine. So now in the bottom panels, we're looking at the impact of updating the vaccine to match the circulating JN1 virus at the time. So here again, starting the preclinical studies on the left, we see that the JN1 vaccine induced about a 10-fold higher neutralization titer compared to the older XBB 1.5 vaccine. And this was also reflected in our clinical studies in the middle, where the JN1 vaccine also induced about this time a 3-fold improvement over the older vaccine formulation. And most importantly, this translated to improved vaccine effectiveness. So we saw JN1 vaccine effectiveness against severe disease in at least one study of 68% during this JN1 dominant period. So overall, these data demonstrate that, by updating the vaccine to match the circulating strains, we can restore and enhance vaccine effectiveness as the virus evolves, very similar to what is being done with the seasonal flu vaccines. So since the vaccine was launched in 2020, there's been 4 approved variant-adapted vaccines, really prompted by the need to keep pace with virus evolution to new variants, as represented by the acquired mutations in the spike protein over time. And these mutations often enable the virus to at least partially escape immune response that's provided by the prior years of vaccine. And through all of these variant adaptations, we also see sustained vaccine effectiveness against severe disease, although the numbers vary depending on study design, cohorts, timing, geographic regions, et cetera. But overall, it seems like although the majority of the population has experience with immune exposure, the virus continues to find a way to escape pre-existing immunity, and it's really by tracking and adapting the vaccine to keep pace with the virus evolution that we're able to maintain vaccine effectiveness year after year. So, with our growing experience with the COVID mRNA vaccine, one of the most important opportunities is now to leverage that insight and apply it to influenza pandemic preparedness. So we can apply the same adaptive mRNA vaccine platforms to more rapidly respond to both seasonal and pandemic influenza threats. Unlike COVID, flu has been around for a long time, so there's already some robust network of laboratories as part of this global influenza surveillance and response system that monitors the circulating variants and detects emerging threats, and continue to strengthen and support these surveillance networks, as well as to invest in advanced prediction tools and algorithms, is key for early detection of flu variants, as well as for the accurate modeling of virus evolution and prediction of predominant strains and immune escape. We can also leverage our lessons learned from our efficient clinical operations during the pandemic and our streamlined regulatory processes to help accelerate clinical validation of new influenza vaccines. And of course, we need to continue building and strengthening our partnerships with both government and global organizations, and continue collaborating with health authorities to establish trust and credibility with these new vaccine platforms. So switching a little bit to flu, one of the key limitations to traditional approaches of making a flu vaccine is that the strain changes or the strain recommendations need to happen months ahead before the flu season actually begins. And that means there's always a risk that the virus will continue to evolve before the vaccines are distributed, potentially reducing their vaccine efficacy. With mRNA vaccine platforms, we have the opportunity to shorten this timeline. So the speed and the technology, speed and flexibility of the technology allows us to potentially reduce the lead time from 6 months to as little as about 100 days, which is what we're currently doing for the COVID strain change. And this allows potentially quicker response to flu evolution and also more agile response to both seasonal and pandemic threats. So at Pfizer, we've been developing a seasonal influenza mRNA vaccine, and this platform has already been validated in the clinic. So we have demonstrated that our first generation mRNA vaccine for influenza achieves superior vaccine efficacy versus a licensed inactivated flu vaccines in adults 18 to 64 years of age in our phase 3 study. Through additional human challenge studies, we've also demonstrated that this pandemic or this seasonal influenza vaccine was able to reduce virus load and also protect against infection with both influenza A and B viruses. And so these data through our seasonal program gives us reason to believe that we can use mRNA technology for influenza to really improve or enhance our ability to respond to pandemic threats. So speaking about pandemics, over the history, a number of different influenza subtypes have infected the human population. But it's this highly pathogenic avian H5N1 virus that has gained the most media attention in the recent years, and it is widely recognized as a pandemic threat. So since it was discovered in 1996, H5N1 viruses have caused over 900 human infections worldwide with a high case fatality rate of about 50%. And globally, the predominant genetic clade is this 2344B, which has driven the largest global avian influenza outbreak to date, with occasional spillovers into mammalian species and the human population. So in the US, we've seen these outbreaks in poultry and dairy cattle, and there's been reports of at least 70 human cases, including 1 death, with 3 cases with unknown sources of exposure. But luckily, most of these human illnesses have been mild, with just conjunctivitis or flu-like symptoms. But the detection of genetic markers of adaptation to replication in mammals warrants continued vigilance and monitoring of these viruses because they will inevitably evolve and continue to reassort. And case in point, just a few days, maybe it's a few days ago now, that the very first H5N5 human infection has been reported in the Washington state. That also belongs to this clade 2344B. So at the beginning of the H5 outbreak in dairy cattle, Pfizer had initiated the development of a pre-pandemic influenza mRNA vaccine that was designed to encode the hemagglutinin, or HA protein, from one of the available candidate vaccine viruses at the time, which is this A-Astrakhan H5N8 subtype that belongs to the same genetic H5 clade 2344B. So this pandemic flu used the same nucleoside-modified mRNA and LNP technology as Comirnaty and our investigational seasonal mRNA vaccines. And in our preclinical studies, it was shown to elicit robust humoral and cell-mediated immune responses in mice. And so as part of this pandemic flu program, we also established a CRADA, or a Cooperative Research and Development Agreement, with the US CDC to focus on the production and characterization of the mRNA influenza vaccines in animal models. So this enabled collaborative studies looking at ferret challenge and transmission experiments, for example, which I'll talk about a little bit in the next slides. But this, CRADA with the CDC, I think really created a share value by combining Pfizer's resources and mRNA expertise with the CDC's expertise in pandemic influenza, their ABSL3 facilities, their assays and reagents to help accelerate influenza pandemic preparedness. And it also helped to generate data that benefits both regulatory and public health goals. So in the next couple of slides, I'll share some preclinical data from 2 ferret studies that we completed in collaboration with the CDC, and then I'll end with some interim clinical results from our phase 1 study. So in our first study with the CDC, we wanted to determine if the pandemic flu vaccine could induce protective antibody responses and also reduce pathogenicity in this lethal H5N1 ferret model. So here we're looking at 3 different groups. So we have the unvaccinated controls, and then we have ferrets that were previously vaccinated with the H5 mRNA prior to challenge. And then we have a group of animals that received an adjuvanted H5 protein vaccine prior to the challenge. So the ferrets received 2 doses of the vaccines 3 weeks apart, and then 5 weeks later, they were challenged with this recent human clinical H5N1 isolate, A. chili, which belongs to this 2344B genetic clade. And then serum samples were harvested at different time points prior to challenge to monitor antibody responses, and then clinical monitoring, nasal swabs, and tissues were then harvested up to 2 weeks after the challenge to monitor pathogenicity. So here we're looking at antibody responses against the vaccine-matched strain as measured by a hemagglutinin inhibition assay. And so as you can see, ferrets that received the H5 mRNA vaccine was able to induce detectable antibody response after a single dose of the vaccine, which was further boosted after the second dose. Whereas in contrast, 2 doses of the adjuvant H5 protein vaccine was required to mount a detectable antibody response. And overall, antibody titers were higher in the H5 mRNA group as opposed to the protein group, and this was sustained up to 7.5 weeks post-vaccination prior to the challenge. And then here's the protection data. So we see 100% of the animals that were vaccinated with the H5 mRNA or the H5 protein vaccine surviving this H5N1 challenge. And on the right, you can see that much reduced body weight losses in these vaccinated groups compared to the unvaccinated controls. And notably, the animals that received the H5 mRNA vaccine was able to regain their body weights more quickly. And so this resulted in higher body weights between days 7 and 14 post-challenge. And here's looking at the virus loads. So on the left, we're looking at virus titers in the nasal wash. And you can see that the H5 mRNA vaccinated ferrets, that's represented by the red bars, had significantly lower virus titers compared to the unvaccinated controls, which were represented by the gray bars, between days 2 and 6 post-infection. And pretty much by day 6, it cleared the virus. In contrast, we still see virus detected in the unvaccinated controls. And then when you look at the tissues, we saw a similar trend, that virus titers in these vaccinated groups were significantly lower than the unvaccinated controls in the trachea and the lung, representing reduced virus replication and shedding in the upper and lower respiratory tract. And we also saw a similar pattern in the small and large intestine, demonstrating broad protection against virus spread. So overall, these results demonstrated that the pandemic flu vaccine could protect against severe disease in these animals and also can reduce virus shedding in key tissues. And this reduced virus load in infected animals could potentially have some implications in limiting transmission. So based on these observations, we conducted a second study in the ferrets, asking whether the pandemic flu vaccine can reduce H5N1 virus transmission in the ferrets. And so here, the ferrets potentially received 2 doses of a vaccine prior to being inoculated, and then 24 hours after infection, each inoculated animal was then co-housed with contact animals. And transmission to the contact animals were monitored by measuring virus titers in the nasal swabs up to 2 weeks after co-housing. So looking at the 4 different groups we tested, with each group containing 3 animal pairs. So the first one is just the unvaccinated controls. So the inoculated animals and the direct contacts were vaccine naive. In the second group, the inoculated animals previously received the H5 mRNA vaccine, and then they were co-housed with naive contact animals. In the third group, the inoculated animals received the adjuvanted H5 protein vaccine and then were co-housed with naive animals. And in the last group, the contact animals had previously received the H5 mRNA vaccine. So for the sake of time, I'm just gonna skip to the punchline, which is summarized in the table below. But you can see that in group 1, in the unvaccinated animals, we saw transmission in all 3 animal pairs. In group 2, where the animals were previously vaccinated with H5 mRNA prior to being infected, we saw much lower viral loads, which translated to partial transmission. So we observed transmission in 2 out of the 3 animal pairs. In the third group, where the animals received the H5 protein vaccine, we saw also a little bit of a lower viral load, but not to the same extent as the mRNA group, but this did not have any impact on transmission, so we saw transmission in all 3 contact animals. And in the last group, where the contact animals were previously vaccinated with H5 mRNA vaccine, we saw that the pandemic flu vaccine could protect these animals completely against infection with H5N1 following their exposure to naive infected animals. So overall, based on these results, with limited numbers of animals, the pandemic flu vaccines shows promise for individual protection, and although it did not completely block transmission, it may help reduce virus spread by reducing virus shedding. So based on these preclinical studies, we conducted a phase 1 study to evaluate the safety and immunogenicity of our pandemic flu vaccine in healthy adults, looking at different dose levels, different vaccination regimens, and also different age groups. But today I'll be focusing really on just these older adult cohorts. So as part of the larger study design, 60 healthy participants, 65 to 84 years of age, who previously received the seasonal flu vaccine over 30 days prior to enrollment were randomized to receive either the 2 doses of the pandemic flu vaccines at 30, 60, or 75 microgram dose levels, or they were given saline as part of the placebo control. And again, the objective was to identify 1 or more dose levels with an acceptable immune response and safety profile in healthy adults. So first, we were interested in looking at the protective antibody levels. So here we're looking at, or we measured hemoglobin inhibition titers across 3 different time points prior to vaccination, 3 weeks post-dose 1, and 4 weeks post-dose 2 against the vaccine-matched H5N8 strain. And what's shown here are the seroprotection rates. So the proportion of the participants that had an HAI titer of 1 in 40 or above, which is based on the established correlative protection for influenza. So as you can see, prior to vaccination, a small percentage of the population had protective HAI levels. But after a single dose of the pandemic flu vaccine, over 85% of the participants achieved those protective HAI levels. And this exceeded the seroprotection thresholds that are established by the FDA for licensure of a pandemic vaccine. And after a second dose, we saw 93 to 100% seroprotection levels. We were also interested in looking at the immune response to a related H5N1 virus that would be more similar to the bovine outbreak. And here again, after a single dose of the vaccine, we saw a dose-dependent increase in the seroprotection rates. And after 2 doses of the vaccine, we again observed 93 to 100% seroprotection rates. So overall, these data suggest that the pandemic flu vaccine can induce a dose-dependent antibody response, and more importantly, we can achieve seroprotection levels that exceeded the thresholds established by the FDA for licensure. We also wanted to confirm our immunogenicity results using a second assay, given some inconsistencies with HA assay for previous influenza strains. So here we were looking at the geometric mean fold rise as measured in antibody titers as measured by either the hemoglobin inhibition assay on the left or the microneutralization assay on the right. And overall, these data indicate that there's a strong increase in antibody titer levels that's dose-dependent in both of these assays. So after a single dose of the vaccine, we saw anywhere from 3 to 11-fold increase in antibody titers, and this increased up to over 20-fold after the second dose of the vaccine in the highest dose group. So overall, the pandemic flu vaccine was also well tolerated at all the dose levels tested, eliciting primarily mild to moderate reactogenicity. And collectively, these data, combined with our experience with the COVID mRNA vaccine and our seasonal influenza vaccine, where we demonstrated efficacy, gives us a very strong reason to believe that we can use or leverage mRNA technology to make an effective pandemic influenza vaccine if needed. So looking forward, we have to start considering or thinking about next innovations for vaccine development, which could include more broadly protective vaccines, especially for COVID and influenza, or looking at alternate delivery technologies to elicit different types of immune responses, like mucosal immune responses that could also help to create preventive or protection and block transmission levels. And so this is an area where I think Pfizer can partner or collaborate with academic and research institutions to really help advance this early stage science. We can leverage, for example, academic expertise in immunology, computational biology, AI and machine learning to really help us in the development of these next generation candidates. And this will allow us also to stay ahead of the virus instead of always chasing the virus and improving our responses for future outbreaks. The second aspect is correlative protection. So I think it's important for us to continue establishing and confirming correlative protection for new vaccine platforms. Standardization of the assays, validating these correlates, identifying new mediators of protection will all help accelerate clinical development and also streamline regulatory approvals and allow these vaccines to be given to a diverse population more quickly during a pandemic situation. And so this is also an area where we can partner externally to help define these correlates of protection for some of our assets. Of course, we need to continue investing in platform technologies, infrastructure, predictive tools and algorithms for future readiness. And and the last point, given the current climate around vaccines, I think it's extremely important to have transparent communication, which is fundamental in fostering public confidence and cooperation during health emergencies. And building this trust with the public and key stakeholders will ensure that all our efforts are not just effective, but also widely supported. And so lastly, I'll just take a moment to acknowledge my colleagues at Pfizer and BioNTech, all the clinical trial participants and their families, all the sites, investigators, staff, CROs for conduct of all these clinical studies, governments and regulatory authorities for their continued partnership and guidance, and of course the US CDC for the CRADA and the ferret studies. So thank you, and happy to take any questions.
Wonderful talk, Pirada. I wonder if anyone has any questions, they can just come to this center mic right here and can line up, and just need a reminder just to turn the mic on when you're about to ask your questions. I wanted just to start to kind of have you maybe dig a little bit deeper into this idea of academia, industry collaborating. I think you touched on some key points there, but I wonder if you could just talk a little bit more detail about where you think are the gaps that academia can fill for industry specifically to help advance your efforts.
Yeah, so a couple of things, right? So just to make it known that at Pfizer, we actively seek partnerships in academia and biotech to really help us accelerate our internal discovery efforts. So we actually have a group that's, it's called our External Search and Evaluation Technology Group. And they're out there looking at different opportunities that would supplement or complement what we're doing internally. So as I mentioned, for more broadly protective influenza vaccines, one of the things is all about translation, translational markers, biomarkers, correlates of protection, trying to understand where or what types of immune responses we really need to trigger in order to have that broadly protective immune response. So again, I said immunology, computational biology, artificial intelligence and modeling, all of these efforts can really contribute to that holistic strategy. Sure.
Tremendous presentation and fascinating. I am communicating a question from my dear friend, Dr. Sridhar, who's a Boston Children's Hospital in the emergency department, and she's like, what about kids? They play a major role in influenza outbreaks as transmitters, and clearly important to develop vaccines that would be available to them and relevant to them and approved for them in a pandemic influenza scenario.
So absolutely, and I think, but in terms of the development, right, we always have to test our vaccines first in adults in healthy populations for some of these disease areas, but there's always an intention to then go down to the younger population. So absolutely, I think both COVID vaccines as well as influenza vaccines are important for the younger population to prevent the serious clinical outcomes.
Kind of following on from that, the flu data for the 18 to 64 age group was, that sounded really exciting.
Yes.
Can you say anything about the over 64 to 65 plus? Is that vulnerable?
Yes. So in that phase 3 study, we showed very strong immunogenicity against the flu A responses. So we showed superior immune responses to the flu A, but we actually missed our statistical non-inferiority endpoint in that older adult age group. But the vaccine was efficacious. It was just from a statistical point, we missed the endpoint there. But as you can see for the pandemic flu, obviously that's why I showed the older adult data for the pandemic flu studies to show that we do get that really, really good, strong immune responses in the older adults.
Quite a few people respond to mRNA vaccines with a general feeling of malaise for 1 or 2 days. Is anything known about the reasons for that, and is there a way to avoid them?
Yeah, so with mRNA technology, part of the mechanism of action is also not just, it induces strong antibody responses, strong T-cell responses, but it also induces some innate immune activation through type 1 interferons. So as part of our next generation vaccines, I think improving tolerability could also be looked at by dose sparing and having more potent vaccines. But
Wonderful, Pirada.
no, thank you.
Thank you so much. We'll move on to our first keynote, our plenary speaker, which is Lillian Juttukonda. So Lillian is a physician scientist studying nasal mucosal responses to viral infections. She completed her MD PhD at Vanderbilt, pediatrics residency at Boston Children's Hospital, and neonatology fellowship in the Harvard Combined Program, and did her research fellowship with Jose Ordonez Montañez. She's now assistant professor of pediatrics at Boston University and an attending neonatologist at Boston Medical Center, where she's building a research program focused on nasal mucosal development in premature infants. So, Lillian.
Hello, everyone. Wow, it's spicy. I am so honored to be invited to be a part of this symposium and just thrilled to share the work that's been a big collaborative project with Jose and many other researchers at Boston Children's Hospital. So looking forward to sharing this. Childhood respiratory infections are still a major cause of morbidity and mortality globally. And as I said childhood for a reason, because although that was really beautiful work talking about seasonal vaccines and COVID and flu, really deaths from respiratory viruses disproportionately affect the very young and the very old. And yet there's a dearth of research understanding, particularly in humans, what drives that susceptibility. So I'm particularly interested as a pediatrician in the youngest patients and what's different about their responses to viruses, and especially the viruses and other agents that cause the most severe disease in young patients, including respiratory syncytial virus. So today I'm going to focus on 3 viruses that are very common in young children. And the first is SARS-CoV-2, which is common, but typically doesn't make young kids very sick. Rhinovirus, which can cause a spectrum of disease, but is particularly well known for causing a wheezing illness in toddlers and causing asthma exacerbations in children and adults. And respiratory syncytial virus, which is the scourge of the pediatric ICU because of its propensity to cause severe lower respiratory tract infection, also called bronchiolitis, in susceptible and very young children. All of these viruses are RNA single-stranded viruses. They all infect the nasal mucosa and then have an ability to spread to the lower airways. But there's obviously different biology that is causing them to have different phenotypes, particularly in young children. So we hypothesized for this project that each of these viruses has a distinct cellular response in that initial site of infection, the nose. And by understanding those differences in responses to infection at that primary site of infection, we might be able to understand some of the differences in clinical phenotypes and then leverage that for treatment or vaccine approaches. So what do we think we know about respiratory viral infections, and what are some of the unknowns there? So a virus must infect a susceptible cell. And for these respiratory viruses, we think that that's the multiciliated cell. But actually, which cells are infected in vivo and the behavior of the infected cell in vivo and how that might differ between different viruses has actually not been well studied. We know decently well for SARS-CoV-2, but not for other respiratory viruses. And while we know that innate immune cells and uninfected epithelial cells can respond to the signal that a neighboring cell is infected, we don't know whether that confers protection and is able to control viral replication, or whether for some viruses, or all of them, whether that adaptive immune and antigen-specific response is required. And in particular, we don't know how this is different between different common respiratory viruses. For the sake of time, today I'm going to focus on questions 1 and 2, which are the infected cells in vivo in young children, and how do those infected cells respond to infection, and what's different depending on the virus. So to ask this question, we built a cross-sectional cohort in the Boston Children's Hospital ED, where babies and toddlers 0 to 4 years of age were enrolled if they came to the ED with respiratory symptoms and tested positive for one of these 3 viruses. They could also be enrolled as controls if they came without any respiratory symptoms for things like, nursemaid's elbow or a laceration. And we collected nasal swabs from each of the participants and then did 10x 3' single-cell RNA sequencing on cryopreserved nasal swabs. And as I said, this was a huge effort. Bruce Horwitz's group in the ED collected the samples, and then Jose, his group oversaw the sample library preparation and data analysis. So I'm really grateful to this whole team that I've been able to be a part of. So we built an atlas of about 360,000 nasal cells, which comprise a great diversity of epithelial cell lineages, as well as immune lineages. And by having this rich data set, we're able to go in and ask a lot of finesse questions about responses, in vivo responses to viral infection. One of the first questions we were interested in addressing is, what is that infected cell? So here on the left, I'm showing just the epithelial cells and showing in different colors the different epithelial subsets or cell states that we identified in our data set. On the right, I'm breaking down those cells to being just those from each of the 4 main groups. And we identified the viral RNA positive cells within our data sets, and we're really interested to see that the SARS-CoV-2 viral RNA positive cells were predominantly in this cluster that we're calling the NF-kBIA ATF3 positive ciliated cell cluster, and that this was very distinct from the RSV RNA positive cells in this cluster that really is halfway between our secretory and our ciliated cell populations that we've labeled the keratin 17 S100A9 ciliated cell population. So another way of depicting this here in a dot plot, you can see that those 2 populations were very distinct for their specific virus. So NF-kBIA holds a predominant high viral load SARS-CoV-2 positive cells, whereas the keratin 17 cells have high viral load for RSV, and that these cell subsets are also specific to that group. So the SARS-CoV-2 infected cells, that subset is only in our SARS-CoV-2 group. The keratin 17 subset that's RSV positive is only in the RSV infected subset. But a subset, the CXCL11 interferon stimulated ciliated subset that had some RNA for both viruses, that subset is seen in multiple viruses. So just showing that we're able to identify really specific cell subsets for each of these viruses. But beyond identifying them, we wanted to understand some of the biology of these cells. So how are they responding to infection? Are there differences between them? And one question we were very interested in is the intrinsic epithelial interferon response. And what we found is that SARS-CoV-2, the SARS-CoV-2 infected cells produce a lot of interferons, so interferon beta and epsilon and interferon lambda, whereas the RSV infected cells, we could only detect interferon lambda. And we were actually really excited to detect interferon lambda in any of these cell types because it's been rather elusive to demonstrate that antiviral response in published single-cell data sets. And it was really interesting to note that the uninfected cells were not producing any detectable interferons at all in the epithelium. We also looked at the transcriptional programs of these cells and said, what's different between the SARS-CoV-2 infected cells and the RSV infected cells? And what I think you can appreciate is that the SARS-CoV-2 infected cells, they are expressing the canonical multiciliated cell markers, as well as multiple genes that are downstream of NF-kappa B signaling, so really showing that there's been a strong innate immune activation of these cells, and multiple interferon-stimulated genes. In contrast, the RSV-infected cluster has low expression of canonical ciliated gene markers, low expression of downstream genes from NF-kappa B, and instead is expressing these genes that are associated with a squamous cell state, such as keratin 17, SPR, 1b. And so we were really kind of surprised to see this and wondered, are these RSV supposed to infect multiciliated cells? So what is the origin of these keratin 17 positive cells? Is RSV infecting multiciliated cells or a different lineage? So to try to address this, we did a lineage analysis using RNA velocity. And in this data set, this analysis, if you follow the arrows, you can see sort of the predicted cell fate. And And it suggests that for SARS-CoV-2, that the precursors are the multiciliated cells, and that this is also true for the RSV positive cells. So this is not definitive, but does suggest that the ciliated cells are the precursor, and that the RSV infected cells, although are now really undergoing a state change and now no longer expressing the ciliated cell markers, and instead taking on these more squamous-like characteristics. So we were really surprised by this finding, and we wondered why and what is the functional role of these keratin 17 positive cells. And so to try to address this, we have turned to an in vitro system, and we have been able to validate in our in vitro system where we infect epithelial cells that were differentiated in culture, infected with a GFP-producing RSV strain, that the cells that are infected with RSV, marked by GFP here, that a subset of these cells are indeed expressing keratin 17 at the protein level. So we're really excited to move forward with this platform to ask more questions about the biology of these cells. Are they allowing, is this a response that is helping the epithelial cell survive infection, providing some mechanical stress resistance, test these types of hypotheses. But in my first slide, I said that most deaths from RSV and respiratory infection don't occur in the US. They occur in other countries around the globe. So we're really excited to have a collaboration with a fantastic research group in Bangladesh, the Child Health Research Foundation. And they, in rural Bangladesh, enrolled 19 participants and collected swabs from them, both healthy and children with respiratory infections. And then on-site in Bangladesh, did single-cell RNA sequencing using honeycomb, a different technology than what we did, and then analyzed this data, including aligning it to a viral genome. And then we were able to take our deeply sequenced large data set and transfer the labels to their smaller data set to ask some questions about what's happening in these children in Bangladesh. So I just want to give a shout out to our fantastic collaborators, and we're really honored to be working with them. So in their data set, they also see a nice diversity of epithelial and immune cells, but what's really exciting is if we look at the RSV infected cells in their data set, again, the cells with the highest viral load and the largest number of infected cells were transferred the label of the keratin 17 S100A9 ciliated cell subset. So they have really a lot of similar characteristics in their data set that we saw in ours. So the RSV RNA positive cells are similar in different patient populations using a different sequencing technology on different continents, which I think really makes me particularly excited to understand what these cells are doing and what that means for why RSV is such a deadly pathogen in young children. So in conclusion, we generated a nasal swab atlas of pediatric respiratory viral infection. And in that data set, we were able to identify unique RSV and SARS-CoV-2 RNA positive cell states. We revealed that RSV infection may lead to a cell state change from a ciliated to a more basal or squamous-like state. And that we identified that this cell subset was also present in locally sequenced samples from our partners in Bangladesh. So in the future, we're really excited to look for correlates of disease severity and age-related responses to RSV infection, and using our ongoing cohorts in the neonatal ICU and pediatric ICU. We're also interested in studying disease progression and disease resolution using longitudinal nasal swab sampling. Interested in understanding more mechanistically what the keratin 17 positive cells are doing and what their role is in viral pathogenesis. And I think that this is a project that really does lend itself to, in the future, partnering with industry. I'm very passionate about the idea of developing vaccines for young children that target the nasal mucosa, and I think that's really important for targeting spread. I think about my 2-year-old in daycare, and you know, it's an exciting place, his nasal mucosa. And with that, I want to thank Jose's lab. It was a wonderful place to train. The last few months I have started transitioning over to Boston University and Boston Medical Center, and very, very grateful to have that opportunity to start my own physician scientist career there. And thank you all for your attention.
Thanks so much, Lillian. I again invite people to come to the center mic to ask questions. Just to kick things off, you mentioned this, we all aware of this difference between a more mild phenotype of SARS-CoV-2 in kids versus adults. If you were to take kind of your data from the nasal mucosa, look at similar data in adults, what are some of the similarities, differences that you observe?
Yeah, so SARS-CoV-2 is probably the best studied virus in the nose. And there actually was a group a few years ago that looked at children all the way through adults. And they said, oh, look, children have this strong interferon response to SARS-CoV-2. They probably just have a strong interferon response to everything. And so they called it like a primed interferon response. And it does seem to be true that we see a strong interferon response in our data set from SARS-CoV-2, not just the infected cells, but also, and I didn't show this today, but there are many, many different interferon responsive bystander populations as well, interferon producing PDCs, so really strong interferon response. And that's not what we see in adults. So there is an interferon response, but it's not as strong, it's not as universal, and it's more blunted in severe cases. So I think that is a difference for SARS-CoV-2, but I don't think it's not that children have a strong interferon response in everything, because we don't see that in the other two viruses. So there's something about, like, children have a stronger interferon response to SARS-CoV-2, and that is still a bit of a why. I don't know the answer to that question. Oh.
Hi. So I've had the advantage of getting to think about this since your talk yesterday, but I'm wondering, in the context of those sort of weird keratin-expressing cells that you're finding in the RSV-infected samples, do you think that a functional consequence of that change in cell phenotype in the infected cells could be a loss of adherence, and that's why they're showing up more prevalently in the nasal swabs?
Yeah, so it's an interesting thought whether they have different adherence, and I don't know if they do or don't. It's certainly something that we could test in our in vitro model. And I can certainly tell you that we have problems when we infect the ALIs, where sometimes we do have peeling of the differentiated epithelium, and whether that has to do with the change in the intermediate filament expression certainly could. And then you have to think, is that a benefit to the host? Is that a benefit to the virus? Maybe that helps with spread. I also have so I think that's a very reasonable hypothesis I haven't tested. In terms of whether, when I say, I mentioned yesterday to you that we see more cells in the virally infected. Mm-hmm. That's not true just for RSV, that's true for the other viruses as well, so rhinovirus and SARS-CoV-2. So I'm not sure if that is a keratin 17 specific phenotype, because I think whenever there's a viral infection, the nasal mucosa is inflamed, it's easier to get cells with a swab. But it's I love that hypothesis for the keratin 17 cells.
It was actually your comment about getting fewer cells out of the healthy samples that made me think about that.
Yeah, great idea.
Thanks.
Thank you for the question.
Hi, I'm Manish Sagar from BU and BMC. It's great to meet you here, even though we're at a relatively small institution. My question was, I'm not a pediatrician, but my sense is that now a lot of kids at birth are getting nirsevimab, and your presentation began with understanding disease severity. So have you had the opportunity to look at whether these keratinized cells are also present? We know even in the presence of nirsevimab, kids are still getting infected with RSV, but they're not progressing to disease. Have you had the chance to look at whether this is a phenotype that's also present in the babies who get nirsevimab?
Yeah, so it's a great question. This data set was 2021 to 2023. So 2023, there may have been a couple, but I don't think we ended up having any in our data set. It was not widely available that season. In our ongoing data sets, though, I think that is a question we'll be able to ask. So stay tuned.
Thank you.
Great, thank you so much, Lillian. So we'll move on to our next speaker, which is Yajuan Wang. So she's a postdoctoral researcher in the Chen Lab at Boston Children's Hospital. She has a decade of experience in virology, and her work spans whole virus structural studies, virus-host interactions, viral envelope proteins such as SARS-CoV-2 spike and SIV-GP41, as well as viral genome packaging. She holds a degree in biotechnology engineering from Zhengzhou University and a PhD from the Institute of Biophysics in China. So, Yajuan.
Good afternoon, everyone. It's really my pleasure to be here today and to share my project with you. First, I want to thank MassCPR for giving me this opportunity. My name is Yajuan Wang. I'm from the Bingchen Lab at Boston Children's Hospital. My project focuses on understanding structural basis of coronavirus genomic RNA packaging. As we all know, COVID-19 was caused by SARS-CoV-2. It's an enveloped positive sense and single-stranded RNA virus. Its genome is quite large, about 30 kilobase, and it is packaged into a ribonuclear protein complex by nucleocapsid protein or N protein. So we can see here is the Korean image of the virus. We can clearly see the major components. The arrow points to spike proteins, and the circle highlights the RNP inside the particle. We have already know a lot about the spike proteins, such as how it binds with the receptor and helps the virus enter cells. We also know much about the replication and the transcription. However, a big question remains. What does the RNP actually look like in the coronavirus? And how does it assemble the long RNA genome? So there is no molecular model for the coronavirus RNP now, and very little were known about how this process works. Nucleocapsid protein has an N-terminal domain, a linker, and a C-terminal domain. When we look at the electrostatic potential map, we see the positive charged patches, which makes sense because they band the negatively charged RNA. The Korea electrotomography data has given us a low-resolution view of the RNP. And when we fit in the domain into the map, suggests about 10 N promoter in per repeating unit. But because the resolution was low, so we still don't know how the RNA was organized or packaged. That's exactly what we are trying to figure out. People have tried to recreate the RNP in vitro, and they do get different shapes like this. And but it hasn't been clear whether these structures are real. For example, a group used a short RNA and got a low-resolution structure, but that didn't look like an authentic RNP. Here is the N protein we purified. So the filtration peak and the band in the SDS page, also the particle shape in the negative stain image shows it's pure. From 2D classes, most particles appear as the homogeneous dimers with a diameter of around about 7 nanometers. Now, when we purify the RNP from the authentic virus, the position of the gel filtration peak and the particle shifts in the negative spin image look very different. 2D classes show a uniform structure about 13 nanometers. So we stored the sample in 4 degrees for 10 days and noticed the circular structure disappeared even though the N protein is present. So to understand why this happened and to check whether RNA is packaged in this complex. So we treated the complex with RNAs. And after the sample treated with RNAs, the complexes were digested quickly, and the circular structure vanished. Well, the N protein is still remain intact, like put it in 4 degrees for 10 days. So these structures show that RNA is really essential for forming the RNP. Here's the image we collected on Titan cores, and the 2D classes show a circular structure with about 10 to 12 promoters. So using about 15,000 particles, we obtained a structure, a map at 9.16 angstrom. And after removing the particles that are not so homogeneous, and then we regenerated a reconstruction at 11.8 angstrom, which fits very well with the Correlate model. So in summary, we have purified SARS-CoV-2 RNPs from authentic virus preparations, and these RNPs contain both N protein and RNA, unlike recombinant N protein. Our CORIAM data shows that these RNP complexes appear to be formed by repeating units that contain 10 to 12 N promoters. So we are now in the process of collecting large data sets to achieve the atomic or near atomic resolution. Hopefully we can get the high resolution structure, and the newly found interaction between N protein or N protein with the RNA will be the potential target for the pharma industry. Finally, I want to thank everyone who helped with this project, Bingchen Lab and Mosan Seed Lab for the virus sample preparation, and also Harvard CORIAM Center for excellent support. Thank you.
Thank you, Yajuan. Again, questions at the center mic. I guess I'll start by just asking, when you look at the RNPs from SARS-CoV-2, do you see anything unusual or distinct? Is it similar to other coronaviruses?
Yeah, that's a good question. When we studied the coronavirus SARS-CoV-2 RNP, because in fact there's no molecular, no very high resolution structure of coronavirus SARS-CoV-2. Even there is the Correlate data published, and they get a low resolution structure, but it cannot tell us how about the N protein and the RNA organized. And in fact, it's really unusual, because in other RNA virus, like the negative sense, because SARS-CoV-2 is the positive sense RNA, and in other negative sense RNA, they always, there's some paper published and study the structure. They always like filament structure. And, but So you have seen the SARS-CoV-2 RNP, it's really like circular shape inside the particle. And this shape never been seen in other RNA virus, positive sense or negative sense RNA. So it's really different. But if there has some oligomer or hair organization structure, it still need to study. Maybe our structure can open the door to understand about coronavirus or the positive sense RNA virus.
Wonderful. Well, thank you so much, Yajuan. All right, we'll move on to our last speaker of the first session. So it'll be Ninad Lasrado. So Ninad is a postdoctoral research fellow and group lead for virology and vaccine research in the Baruch Lab at BIDMC and Harvard Medical School. He develops thermostable RNA and adenovirus vaccine platforms for mucosal delivery and a focus on accessible global deployable vaccines. His research spans mucosal immunology, AI-guided vaccine design, and pandemic preparedness, building on prior work in viral vaccinology and immune diseases. So, Ninad.
Hello. Sorry. Well, good afternoon, everyone. Oh, still. All right. My name is Ninad Lasrado. I'm a postdoc with Dr. Dan Baruch at BIDMC. And I want to thank Nadina and her whole committee for giving me the opportunity to speak about the development of a clade 2.3.4.4b H5N1 avian influenza vaccine that we especially look for mucosal protection in multiple animal models. Just going to briefly give some introduction about influenza A viruses, which is what I'm going to talk about. It's a negative sense RNA virus, which is made up of 8 genomic segments. And the viruses are classified based on the surface proteins, which are the hemagglutinin and the neuraminidase. And there are different subtypes of the hemagglutinin. There are 19 subtypes of hemagglutinin and 11 subtypes of neuraminidase. And there's different combinations of hemagglutinin and neuraminidase. But the interaction of the hemagglutinin with the sialic acid receptors, that's the major determinant of host range and tissue tropism. And today I'm going to talk about H5N1, and Pirada already spoke about it, so I'm not going to give a lot of background information. But just briefly, the origins and spread of H5N1, as Pirada mentioned, in 1996, we saw the first human case, as you can see with the blue dot over here in China, where the first case was identified. And over a period of time, you do see this spread, spill over into mammals, spill over into humans, all across Asia, and by 2014, 2019, you do see this spreading over the northern hemisphere, mainly by the migration of wild birds, which carry this virus and infect seals or other avian populations. And in 2020, between 2022 to 2024, we did see that this has almost spread all over the world. And speaking about the US, we did see the first poultry outbreak in 2022. And this is directly taken from the USDA website, which shows the spread of H5N1 not H5N1, just HPAI, in the poultry population. And it was first detected in February 2022. And there have been more than 1,500 flocks that have been affected. And unfortunately, more than 200 million birds had to be culled. And it's been devastating for the flu industry. Not flu, sorry, I don't know why I said flu industry. It's not devastating for the flu industry, but it's devastating for the poultry industry. And all 51 states and territories were affected, as you can see here. But what was more interesting in 2024 and surprising for some of them was that there was a spillover from birds to cows or cattle, and this was a major shift. You saw the reservoir shifting from birds to mammals, and this was a cause for concern. And we did see up until 2025, I guess in March 2025, we did see close to 1,000 cases of the herds all over the US in 17 states. So this was a little concerning. And our lab is a vaccine research lab, and we study viral immunology and also bacterial immunology, and we develop countermeasures for such pathogens. And the first objective that we had was to develop nucleoside-modified messenger RNA vaccines and rhesus adenovirus vectored vaccines against the outbreak strain of H5N1. And the second objective was, once we developed these vaccines, we wanted to evaluate the immunogenicity and protective efficacy in mice, chickens, calves, and non-human primates. So we wanted to have a big picture about how these vaccines work. And as a typical vaccine research lab does, we generated these different vaccines, and we started with the mouse model. And we wanted to test how the different gene-based vaccines work in mice. We tried to give the hemagglutinin HA, H5N1 vaccines, be it mRNA or rhesus ad vectors, intramuscularly or intranasally, and we followed up to 12 weeks with intermittent blood collection and mucosal sample collection to see which platform induces good systemic and mucosal immune responses. And as you can see here on the left-hand side, it shows the serum IgG responses against the H5 hemagglutinin. We do see over here in purple, that is the mRNA vaccine. Over here in green, that is the rhesus Ad52 vaccine given intramuscularly. And this is the rhesus Ad52 vaccine given intranasally. But we do not see responses against the mRNA given intranasally, largely owing to the biophysical drawbacks of the LNPs, which do not penetrate the mucosa. And we do see these responses, they're more comparable and stabilizing over a period of time in every group except the intranasally given mRNA group. And the serum neutralizing antibodies we evaluated using a pseudovirus-based assay that we developed in our lab, and it showed a similar picture where rhesus ad vectors given mucosally are systemically, and mRNA given systemically induce good peripheral neutralizing antibody responses. We next wanted to see how this translates at the mucosal level, and we developed a protocol where we collect nasal washes without killing mice, where you serially collect nasal washes, and we did see that in the RAD52 intranasal delivered mice, there was an increase in immunoglobulin A. And similarly, at termination, we did see higher levels of IgA in the BAL of these mice. We next looked at the tissue-resident memory T cells, and these T cells are very important to kill any virus-infected cells at the site of seeding. And we did see substantially higher levels of tissue-resident memory T cells in the nasal turbinate, which is the upper respiratory tract, and the lungs, which is in the lower respiratory tract. So based on all of these, and keeping in mind of the One Health approach for veterinary purposes, we also tested our rhesus ad vectors intramuscularly and intranasally in chickens. And this was done with a collaboration with my alma mater at the University of Nebraska-Lincoln, where we vaccinated the chickens at week 0 and week 3 and looked at peripheral and mucosal immune responses. So here we looked at only RAD52 because that's the most feasible approach for chickens. We did look at serum antibody responses, and as you see, with intramuscular vaccination, you do see robust serum binding antibody titers. And also with the intranasal vaccination, you do see post-boost these serum antibody titers coming up. And these are preliminary results. These are not published data. We're still looking at other samples. And we also collected nasal swabs from chickens. And we did see that there was an increase in IgG mucosal responses in these vaccinated chickens, and superior IgA production post-boost in the chickens which got the RAD52 intranasal vaccines. And then we wanted to see if these gene-based vaccines, right, can we test them in cattle, because this is another reservoir, and it's an important finding. And we thought we could test mRNA vaccines and RAD52 vaccines intramuscularly, because intranasal administration is a little difficult in cattle. And again, preliminary data, we do see robust serum binding antibody responses against the H5 hemagglutinin with both mRNA intramuscular prime boost and also RAD52 prime boost. So these all these data were very encouraging for us, and we wanted to design an optimal study to translate this to humans in case there's a human outbreak. What do we do, right? So we conduct a lot of non-human primate studies for translational immunology, and based on all these findings, we were able to design a study in cynomolgus macaques where we vaccinated monkeys with these nucleoside-modified mRNA at week 0 and boosted them at week 8, they received RAD52 vaccines as a prime boost approach intramuscularly or mucosally, and 9 weeks later they received an H5N1 lethal challenge through the intranasal and the intratracheal route, and this isolate was from an infected human who had conjunctivitis. So we wanted to give a really human translational significance for this challenge, and 2 weeks later we necropsied the monkeys. So in all these figures, I'm going to show you some data. On the top panel, you'll see the H5 Texas, which was the outbreak strain, and H5 Astrakhan in the bottom panel, which is a historical H5 strain that's used in the vaccines as well. We do see a robust increase in serum IgG responses post-prime, and robust increase in these serum IgG responses post-boost. It doesn't matter if it's mucosal boost or a systemic boost. We really do see good serum binding antibody responses. And we wanted to next look at the mucosal immune responses, and the IgG. So in the BAL compartment, we did see a robust increase 2 weeks post-boost, mucosal boost, a robust increase in IgG responses with both the H5 Texas strain and also the H5 Astrakhan hemagglutinin. And these, the trend continued until the termination of the study. And we wanted to look at IgA responses, and IgA is very essential to neutralize the virus. And as you can see over here, just the RAD52 mucosal boost induced robust IgA responses. And these will correlate later when I speak about how it suppressed the viral loads. Next, we looked at the upper respiratory tract, the nasal swab IgG responses, and although these were not statistically significant, we did see a higher trend of IgG responses with the mucosal boost, as you see in the blue dots over here. But similar to the BAL IgA responses, we did see an increased IgA production in the upper compartment. We then looked at the neutralizing antibody responses, systemic and mucosal, but I'll go for systemic first. We do see post-prime increase in or some levels of serum neutralizing antibodies that get boosted 2 weeks post-boost. They go to a higher level and somewhat stay similar and do not wane over a period of time. We then looked at the BAL and the nasal swab neutralizing antibodies, similar to what we saw with the nasal swab and BAL IgA responses. You do see an increased trend of higher neutralizing antibodies in the lower compartment of the respiratory system that persist over a period of time. And similarly, we do see an increased trend for nasal swab neutralizing antibodies. We did look at cellular responses at the systemic level. I'm not going to show that here, but just to focus on mucosal immunity at this point, we looked at the tissue-resident memory T cells, both CD4 and CD8 tissue-resident memory T cells in the BAL. This was week 10, so 2 weeks after boost, and we do see a significantly enhanced tissue-resident memory T cell population for both CD4 T cells and CD8 T cells, which was very fascinating for us. And based on all these results, we next wanted to do a challenge, as I mentioned earlier, with the human Texas 37 strain, and we did see that all the vaccinated macaques survived the lethal challenge, which is always what we look for in a vaccine study, protection from severe disease and death. And almost 80% of our macaques which did not receive a vaccine succumbed to death within 5 to 7 days. We then looked at the viral loads in these macaques, and as I mentioned earlier, there were no viral loads in the periphery or in the plasma of these vaccinated monkeys, but we do see that in monkeys which did not receive a vaccine. And more importantly, looking at the BAL in the nasal swabs, we did see higher amount of viral loads in monkeys which got the vaccines intramuscularly. Didn't matter which platform it was, there were higher viral loads when vaccines were given intramuscularly, but we do see a shunt in the viral loads which resolve within 2 to 3 days in monkeys which receive the vaccines mucosally. Similarly, in the nasal swab, we see the same trend where there is a lower viral load compared to the sham vaccinated macaques, but when you give a mucosal vaccine or a vaccine given mucosally, you do see a blunt in these viral loads. And just to give a better picture of the two viral loads, this is how it looked like. In the BAL, the viral loads were at the log 3 level, whereas in the mucosal vaccinated monkeys, whereas in monkeys which received the vaccines intramuscularly, they were at the 10 raised to 7 or 10 raised to 8 level. This shows that the mucosal vaccination is really robust in preventing viral dissemination. And to summarize, we developed H5N1 hemagglutinin messenger RNA and RAD52 vaccines, which are immunogenic in mice, chickens, cattle, and non-human primates. And the mucosal boosting with RAD52 vaccines induced robust systemic and mucosal, humoral, and cellular immune responses. These provided protection against severe disease and death. And mucosal boosting also resulted in enhanced virologic control compared to monkeys which got the vaccines systemically. And these gene-based vaccines can be used for both veterinary and human purposes. And I want to mention that we work with Pirada and her group. We published a paper earlier this year on developing or alternative vaccine platforms or delivery strategies, and we were able to show that for seasonal vaccines, when vaccines are given mucosally, you do see enhanced protection. And I believe that should be the approach that we should take, good industry collaborations and also targeting alternative vaccine delivery platforms, target the mucosa for respiratory diseases and things like that. And I want to acknowledge everyone, starting with Dan, who's a great mentor and who's been very supportive with all these projects. I want to thank the whole lab. We are a very big lab, but it's been a highly collaborative project. Interesting collaborations, interesting discussions, different groups within the lab, and people at BioQual who connected all these large NHP studies, and of course, people at the University of Nebraska-Lincoln who conducted the chicken and cattle studies, and MassCPR for funding part of these studies. Yeah, I'm open to any questions. Thank you.
Wonderful talk, Ninad, and again, open for questions. I'll just start with one. One of the concerns around adenoviral vaccines has been this vaccine-induced thrombocytosis and thrombosis and thrombocytopenia, which is rare but can be quite devastating. Curious your thoughts about adenoviral vector development for vaccines in light of that complication, and also about intranasal delivery and any concerns that with respect to that complication and that delivery modality.
Sure, great question. So I think the answer for that is in two parts. First of all, when it comes to the RAD52 vectors, they have a neutral charge on their capsid, so they do not bind to PF4, whereas at 26 ChAdOx, all of them had a net negative charge. So binding of these capsids and the hexon region with the PF4 resulted in these complexes and some unfortunate deaths. But with RAD52, we do not see that. The other thing with the mucosa, you don't have a lot of PF4 over there. You see an abundance of PF4 in the systemic region. So intranasally, based on both of these factors, I think RAD52 vectors would be really helpful. Yeah.
Great. Okay, so we'll begin our second session. Hope everyone enjoyed the break. So I'm going to introduce our second keynote speaker, and that's Régis Gervier. So Régis leads Sanofi's mRNA Center of Excellence, an integrated organization advancing mRNA prophylactic and therapeutic solutions across research, LNP technology, CMC activities, GMP operations, and applied data sciences with teams in the US and in France. Over his 3-decade career at Sanofi, he has held global R&D and industrial leadership roles across Europe and the Americas. Before establishing the mRNA Center of Excellence, he led the global Sanofi Modulus program, which is recognized by Time as a 2025 Best Invention, which revolutionized biologics manufacturing and was implemented in France and Singapore. He holds a biotechnology engineering degree from Polytech Marseille and a master's in advanced microbiology and an MBA from McGill University. So my pleasure to introduce Régis.
All right, can you hear me well? Just checking. Okay, perfect. So very, very pleased to be with you today, and thank you very much for the organizer to invite me to speak on behalf of Sanofi on what we are doing actually, globally speaking, in order to innovate all along the value stream for vaccine development, launch, but also addressing pandemic preparedness. I picked the title of Innovating for Better Outbreak Interception because you're going to see that basically we are really innovating at scale all along the value chain, using notably AI, machine learning, and that's one point I would like to focus on today in my talk. So rapidly, just to introduce Sanofi for the one who don't know it, it's a French company, right? But very, very well established in many countries, notably in R&D, but also operationally speaking. You see the number, it speaks by themselves. Very proud of the fact that we were talking about the RSV. We have already immunized 6 million infants with B. fortis, basically. And we are very, very proud of the vaccine GBU that we are already covering 500 million people every single year with our vaccine. Very much focused on innovation. I talk about the RSV, but I will come back to it. On influenza also, that we are continuing to invest on the different platforms that you're going to see just after. On the vaccine side, globally speaking, worldwide leader in influenza and pediatric vaccine. You see the number. Very proud of what we are doing on polio vaccine too. As you can see, the number of doses distributed worldwide. And we have actually the best and the biggest capacity of production of our polio in France currently. And I already mentioned the number of million people we are touching every year. What is super important to understand, if you want to innovate at scale, not only you have to innovate on new platform, but you have to evolve also your legacy platform. And that's the beauty of Sanofi today. We have a chance to have decade and decade and decade of development of vaccine for flu, seasonal vaccine. And we are leveraging this ecosystem and this mechanism of improvement on a daily basis in order to bring new solution to the patient. And you can see progressively, but surely along the years, that we move from egg-based technology for seasonal vaccine, but also for pandemic vaccine, up to recombinant protein, thanks to Flublok and the acquisition at that time of protein science. And more recently, with Novavax partnership, being able to really bring a combo vaccine based on recombinant technology. Last but not least, mRNA technology, we were a little bit behind others potentially in 2021, and you will see that we catch up. We enter in the race, we invest massively on this technology. A lot of things to do on it, I will talk about it, but many, many progress that have been done. So the mRNA Center of Excellence, the numbers are not up to date. It was probably 1.5 years ago, but globally speaking, the message is the same. We build an accelerating engine, and it's very important to be able to do that with an end-to-end organization, which cover, notably through the acquisition of Translate Bio here in Boston, all the research and development part on the platform itself. And after the late development industrialization asset that we have in France, allowing us to launch at speed any given target we want to focus on through the mRNA techno. In 2.5 years, we hire roughly 350 people, very young talent from university, academic, biotech, out of PhD and postdoc, but also coupling them with a little bit more seasoned people like me, in order to be able to orient a little bit the research, but to make sure that we are taking the right level of risk to develop any given product in the pipeline. So also importantly, and this is something that you have to understand, we have more than one century of vaccine development in Sanofi history. And therefore, we have a sum of platforms, you can see on the left-hand side, that allow us really to select the best platform for the disease we want to target. And we are considering today mRNA vaccine, and we know perfectly that it will not work for every single disease. And therefore, we are really leveraging this arsenal toolbox in order to feed our portfolio. Portfolio has been reshuffled also recently in order to really distinguish 3 different buckets of product. One, which is maximize. It's basically, you can see on the right-hand side, where we are leader today. We are continuing to invest, notably introducing mRNA technology in this portfolio. We are expanding on the respiratory disease, notably on the middle part of the cartoon. And we want to pioneer leveraging mRNA technology also on some disease and some target mechanism of action that are super complex to crack, like, for example, acne, chlamydia, but also a lot of more that we cannot disclose today. But just to give you an idea, we do believe that today mRNA technology will represent 60 to 70% of the overall portfolio of the vaccine in the future. Now, we were talking about flu. We are leader in flu, definitively. Here you have very recent news that we communicate to investors, basically 2 or 3 weeks ago, with a very, very important study, phase 4 study on flu HD, flu adults, commercially speaking in Europe, where you do see that through these studies, the flu vaccination impacts significantly the health of the people, and notably all the comorbidity, with a strong reduction of pneumonia and influenza hospitalization on one side, but also laboratory confirmed influenza hospitalization, which is very important to communicate to the full population in order to make sure that they get their flu shot every single season. We also expand the application of fluconidose recently from 65 plus down to 50 to 65 years old, with very, very strong phase 3 results. And as we said, flu pandemic is definitively at the core of our agenda. The advantage is that we are in it for a while. We have different platforms, be it eggs, be it recombinant technology, but also mRNA now with the phase 1-2, and we were talking about it just before, with very, very strong results in our hand with our mRNA techno. We have also signed a new phase 1-2 collaboration with BARDA in order to test adjuvantation with the famous adjuvant that is coming with a Novavax collaboration. Last but not least, flu and COVID combination that we are bringing also to the table with a very, very strong positive phase 1 and 2 results. Now, when you are talking about making sure that you are ready for pandemic, you need to think about science, and we heard about a lot of science already in the first session. But you need also to be ready on the manufacturing side. And I will give you a little bit some insight, notably on the design of the facility we have put in place in Sanofi, which is a kind of flagship model that we are replicating in every single country, but allowing you to produce 4 or 5 different antigens in the same room without specific issue in terms of regulation. Last but not least, the notion of maintaining the ecosystem warm, being from surveillance, but also research and development at full speed. So late development, the tech transfer, and also the full education of the population practitioner in order to make sure that we can maintain a high level of coverage rate. So a little bit a chapter on mRNA. Where are we coming from? You know, sometimes there are some advantages to start a little bit later than the others. Is that you know already the drawback before starting. Therefore, you can focus a little bit more on where you will make the difference. So what we did in order to start at full speed in mid-2021 is to go full-fledged. Here there are only 3 different LNPs with different, as you can see on the left, configuration of mRNA, but also multivalencies, different target, different LNPs. We wanted to really calibrate, according to the LNP we were using, what is the immunogenicity obtained for a given target, but also the reactogenicity. That's what you see on the right. And you can see that according to the LNP you use, you can see totally different results and totally different behavior. And this helps us notably, I know that there is an animation, to target an LNP that allows us to have very high immunogenicity but very low reactogenicity. To do that, you need to work on all the dimensions that are on this slide. At the beginning, when I start on this technology, because I was more coming from the small molecule world, but also monoclonal antibody and vaccine with classical platform, we were really focused on the delivery techno itself and the mRNA design. You see it here. You have all the elements that are entering in the game. But you have also the CMC part that we have a tendency to undermine in terms of impact on the final immuno. And we are doubling down on every single axis to make sure that at the end of the day, we have the best-in-class platform. This cannot be done without AI machine learning, definitively. When we hired the 550 plus people of the center 2.5 years ago, we put 10% of the workforce on AI machine learning. If you don't have that, you are nowhere. And therefore, we applied it along the value chain, starting with mRNA design, and I will come back to it, LNP design and selection, translational science, and manufacturing process performance. Because according to the sequence you're going to put in your process, you will not have at all the same performance at the end of the day in terms of quality and yield, and therefore doses for your population. So an example of what we have done, and we published recently in Nature Biotech, we developed several algorithms, generative, predictive one, and we also developed deep neural network in order really to predict the overall performance of our construct. Here we are talking about the coding sequence. We can also improve the non-coding part, giving you an example of how you can improve definitively the translational rate, but also the overall protein production with an optimized sequence. A little bit more classical, but we design a hybrid model for the CMC part. The point being, as soon as you put your sequence in the reactor, you have to make sure that at the end of the reactor and the purification, you have exactly the same quality, whatever the target, whatever the sequence, whatever the multivalent component you want to produce. So it's something that we progress a lot on, and now we are progressing more and more. The next step will be a full-fledged digital twin, meaning that just in silico, we'll be able to predict the performance of our process to make sure that we can also optimize the occupancy rate of our facility. If you go a little bit further, I was talking about translational science. We have also equipped ourselves definitively with what we call our Mimic platform. It's something that we have in the organization for more than 15 years, I think, Josh, right? Just by acquiring Vax Design in Orlando. But recently, we were talking about collaboration, right, with academic group, university. We developed an ecosystem in Australia in order to make sure that we can go very fast in clinic in human for translational studies, and really understanding better the overall dimensions that are mentioned on the right of the slide, understanding better the mechanism of action, focusing on immunomodulation, and have a better understanding of host-pathogen relationship, notably in the frame of chlamydia, because we know that in Australia, notably in the koala population, they have a very good understanding of the disease and this type of bug. Now, what does it take in terms of predicting reactogenicity using machine learning? So there is a forward translation, which is to say you use an in vitro system mimicking urinate response. You try to correlate some cytokine and signal with the different side effects that you can observe, being fever, chill, headache. And through the model, you start to classify the different formulation you can have. And you can see very well here in the middle, with unmodified RNA, you see that globally speaking, you have more reactogenicity than modified. Sorry. You can see also that you have some variances with the different LNP that you put in the system, using our control as Flublok, which is a recombinant protein vaccine. I'm happy to say that as we pursue down the road the overall trial, we have been able to validate the model with actual human trial, showing exactly the same classification of the LNP versus the preclinical model. If you want to enrich this model, you have to reverse translate, meaning that you use your clinical population, you use also the PBMCs of the vaccinees, and you look side by side your cytokine signal between human and in vitro, and you can fine-tune your model in order to make sure that you enrich the robustness of it, and that you really can predict what will be the reactogenicity of your formulation of your vaccine down the road. What we are doing also, and I think we probably, we talked about it already a little bit earlier, we want to look how can we improve the breadth of protection of our vaccine. And here it's an example of looking across the space, what type of strain sequence we could put to enhance the seasonal vaccine, for example. Here it's preclinically in a ferret model. As you can see on the right-hand side, the gray dots, if I am correct, are the WHO recommended strain, is a WHO recommended strain. And on the right blue side, you see the additional strain that we propose to put in the vaccine in order to increase the breadth of protection. This is preclinically. If you look clinically, so we ran a translational study, and you can see the population here, 18 to 40 and 65 plus. And we are demonstrating, if you look the graph, the WHO recommended strain is basically the green line, and you look the space across the different heterologous strain we are testing against. We had a control where we double the dose just to make sure that we can compare side by side an augmented version of the seasonal flu versus the classical one. And we put also H3N2 WHO plus the machine learning strain selected. And as you can see, we are really increasing the breadth of protection for the different strain against we were testing. Last but not least, using machine learning, structural-based antigen design. I mean, Pirada talked about it also. Very important to have this tool in order to make sure that for every single antigen we target, we have the best construct to stabilize the antigen in the prefusion form. And it's something that we are leveraging this here on H5, but on HMPV, for example, that we are putting in the combo vaccine with RSV. And it allows us really to reach the best performance in terms of overall response. So this is pure science, but as I was telling you, pure science, if it stays like that, is not worthwhile, because you need to make sure that you are equipped also to go very fast to the market, and you are equipped to react very quickly in case of pandemic or outbreak. And that's what we invent, if I can say so. First of all, we have, as you can see on this slide, a very, very large network of manufacturing capacity with historical technology like eggs, as you can see in Canada, in the United States, in Swiftwater, and also, I don't know if it's mentioned here, but yeah, Mexico and China, where we are really the largest egg capacity production, vaccine-based production. And we have the famous new facility I was mentioning, notably for mRNA technology, but all other technology that you can put in this room, which are based in France. That's the first plant that we build, and we are currently qualifying, Neuville-sur-Saône, right? And we have also one in Singapore, which is a twin of the facility. The idea being able to pilot at the end of the day the two facilities from the same space or place to really have this twin notion in place. So what is this facility about? It's something that I designed during COVID, by the way, where we start from an idea to say we have to completely stop the model we were used to use in Sanofi to produce biologics, and we want something that is the most flexible possible on Earth. And for that, you have to forget all the things you have done in the past, and you have to educate authorities, FDA, EMEA, and MHRA, etc., etc. The idea is to put several levels of modularity in the system. You put modularity at the level of your building, so you can plug additional, what we call ballroom for reduced cost. You can do it in 2 years instead of 6. It's huge use facility, and every single ballroom is 100-meter length. And after we come with standardization, a little bit like in the automotive industry, in order to make sure that we can select pre-established technology, whatever the vaccine you want to produce. It's a kind of Lego. You just play with the articulation of them in order to make sure that you can produce at the end of the day. Be it AAV, be it recombinant protein, CHOP, E. coli, yeast, whatever. Once you have standardized this, you use maximum standardized single-use, standardized equipment model, and the last point, what we call mobile plug and produce, is basically we went to Audi and Porsche, Volkswagen Group, to understand a bit more how they configure their production line, in order to make sure that they can switch from one model to another. And we apply the same concept in the pharma world. And last but not least, in terms of if you want to go fast, you put a digital twin on your facility, but also on your process. And we are progressing on the notion of self-healing process, making sure that a process, as soon as you put your seed or whatever in the reactor, is auto-correcting its trajectory. You have pictures of the two facilities, the one in France, which was a brownfield, meaning that we were capitalizing already on a facility existing for the utilities and so on. And in Singapore, a brand new one out of a greenfield that allows us also to accelerate the launch. Last point, very, very important, it's making sure, I'm okay on time? Yeah. Last point is really, as we were saying, how can we make sure that the full ecosystem stay warm? You know, it's like when you are an athlete, you have to make sure that you are ready to accelerate the day you have to do it. And this is coming for sure with, it's all about us in science, but it's all about the ecosystem we are piloting and we are collaborating with outside, notably with authorities, but also practitioners. And when you take flu, we were talking about pandemic flu, in order to address a pandemic tomorrow, we have to make sure that the population, the practitioners are ready to jump on it. And what is a little bit a paradigm that we have to address is linked to seasonal flu. And it's an illustration that we can use in order to prime a little bit of reflection. First one, seasonal flu is a vaccine that is available for decades. And I would say the health system are very well educated and trained in order to make sure that we can really reach the full population. However, people have a tendency probably to forget a little bit the impact of this disease that you can see here on this slide, impacting dramatically the population at the global level, but also in the US definitively, with still a lot of cases of illness and respiratory deaths worldwide. And despite this burden, when you look at the overall coverage rate that you observe globally speaking, okay, it's satisfactory, but not sufficient. Only 75% today of target that WHO is putting on the table, and you can see on the right-hand side, definitively a progressive, I would say, decrease, even if it's not huge, but we see that there is an hesitancy, definitively, to go to the practitioner to get your flu shot. And this is a paradigm that we need to revert, definitively, because the impact of this between the disease impact itself, the reduction of the vaccine coverage rate, you have a big, big impact on the overall supply that we have to produce and to give to the different population. You see it on the left-hand side, definitively. After COVID, there was clearly a boost, but progressively, but surely, we do see a decrease of the overall doses distributed worldwide. And this is also visible significantly in the US, as you can see, moving from 194 million doses to 148 today. So there is really an equation to crack here. Particularly because we don't know what will be the next pandemic. Even if we believe, and logically it should be probably a flu pandemic, we don't know what type of magnitude this pandemic will come with. It could be a very mild one, like in 2009, as it can be moderate, like we had with COVID, leading to a quite substantial amount of deaths and the requirement to have a lot of million doses distributed in a record time. Pfizer did a great job to do that, definitively, with BioNTech. But also, we have some cases where it can be even more dramatic. So we need to be ready for that, right, altogether. And to do that at all level, definitively, Sanofi level, we have, as I was mentioning, the different pillars that we are contemplating. The first one is really making sure that we collaborate even more with the patient community, healthcare provider, in order to prime them and making sure that there is very, very strong education on the immunization campaign. We have also to leverage all the technology we have. Eggs definitively is the cheapest. We have huge capacity in Sanofi in order to answer to a pandemic with this technology. However, you have even better with mRNA, definitively, because mRNA can be even faster, a faster technology to put on to reach the market, and the simplest one, definitively, compared to eggs techno. Last point is the mature network of manufacturing. That's what we have in Sanofi today, leader in eggs, flu-based vaccine, so we can move from seasonal to pandemic vaccine, and we are building the capacity on mRNA progressively but surely in order to answer any specific needs. So in a summary, as we were saying, definitively, if we want to succeed, we need to improve the immunization rate. And it's not only Sanofi or private companies that can do that. It's a full ecosystem, collaboration with government that can give the direction, and us behind the scene, making sure that we are ready to jump on it and execute right the first time. We have to make sure that we can maintain the facility warm and the workforce warm. This is quite easy with eggs, basically, because you have seasonal vaccine. You can switch to pandemic, for example, today, at least in Sanofi. With mRNA, it's even better because we are using mRNA for the full portfolio. As you have seen, RSV, HMPV is one of our targets, acne, chlamydia. So we have a facility, and we will have facilities that are going to run every day with mRNA. So you just have to change the sequence, and you are good to go. So very, very important to have that in mind. We need to continue to invest or invest even more on public-private partnership in order to make sure that everybody understands very well the immunization ecosystem to increase the vaccine uptake. As you have seen, not sufficient today. People are losing the reflex to go to the healthcare provider for their shot, and we need to continue to educate here. Global collaboration and coordination is a must to accelerate availability of vaccine. You were talking, Pirada, about confidence, credibility, trust. Definitively, we need to insist on real-time predictive surveillance to anticipate and manage epidemics and pandemics. The notion of regulatory, global regulatory uniformization is important. You know perfectly, if you take monoclonal antibody on one side, you have only one monography for a worldwide application. If you take vaccine with all the platform we have, all the legacy we have on it, we have 50 different monography. And this cannot continue. We need to make sure, and using the approach with the mRNA platform, we have to make sure that we can leverage this approach. Last but not least, we have to foster really focus on communication, very simple, pedagogic, transparent, so that people can gain back trust on vaccine and vaccinology, and also improve the overall coverage rate. So we have an opportunity, and I think Pirada mentioned it very, very well. Listen learn from COVID. It's all over the place. We can leverage that and making sure that this type of situation will not reoccur. So you were talking about, and you know that you're going to ask me this question, so I will answer already in advance. What are the key elements that we should focus on for collaboration? So there are different approaches. So if you look at the mRNA platform and the way we manage it in Sanofi, definitively we know perfectly that doing only research internally will not make us the leader in this techno. We need to collaborate. In 3 years, we signed 150 MTA, just for information, and we have 50 different partnership collaboration, either with private company, but also with academics, in order to crack the code of all the elements I was mentioning at the beginning, be it mechanism of action, design of mRNA, LNP pharmacology or pharmacodistribution, etc., in order to make sure that we can really inform the full population and ourselves internally on where can we use mRNA and where we should drop it, as it's not the best technology for the target we are contemplating. Here you have some example. I was talking about Griffith University, Australia, translational hub, also very much linked to chlamydia disease. So it's an opportunity for us to be in this ecosystem. We are currently collaborating with Singapore also for acne, because they have a very strong center on skin research, etc., And you see the different collaboration we have in the US, but I'm pretty sure there are more to come, definitely. So I like this sentence. Typically, when I start this journey on mRNA, but also seeing what happened in COVID and post-COVID, I have every single morning this type of slide in front of me in my office, which is to say that everyone here has a sense that right now, it's one of these moments where we are influencing the future. And clearly, this young generation in the room, and the less young, the senior who are guiding the youngest, are here to make it happen. Thank you very much.
Thank you, Régis, for the wonderful talk. I can encourage people to come up and ask questions. You did take my question about industry and academia
not completely. You still have one part.
But actually, I want to ask about the modulus system. I think it's just such an innovative approach. I guess if you could distill down, what you think are the key aspects or the key learnings there in preparation for the next pandemic, what would that be if we were to think about kind of key concepts for preparedness?
Yeah, basically modulus was coming from the fact, and I don't know if you have that in mind, but the vaccine industry 10 years ago was an old-fashioned industry. And so we were using pathogenic strain or strain in order to produce antigen, and this triggered a very complex manufacturing network where you need actually to build a facility specific to the antigen you want to produce. When you are in a pharma industry, the difficulty with that is that commercial colleagues may not have completely strong accuracy of what will be the market overall of a given product. So you end up with facilities that are not completely occupied, cost you a lot of fixed costs, and are not very flexible if you have to switch over from one technology to another or from one antigen to another. So that was the idea a little bit of modulus at the beginning, to say if we want really to be fast and to go in 3 or 4 months from nothing to production, or even do a turnaround time, you're changing your production line, in modulus it's 7 days. If you're asking me a recombinant protein now, I can do it in 7 days. If you're asking me mRNA tomorrow, I just unplug my equipment, plug the mRNA equipment, and in 7 days I can turn around. So it was all of this concept to say, okay, if we want to be agile, we need completely to disrupt the model. And it helped after, and that's what we are observing today, to take more risk at our level because we can build facility without knowing what we're going to put in it. Still, we need to be sure that there are activity in it to absorb the fixed costs. And that's where this notion of having platforms that are used in the commercial space, but also in the pandemic space, are super important to keep the people trained, to keep the equipment running, and to make sure that we are not asking government to pay fixed costs for an empty facility at the end of the day. And maybe just before we go there, just to finalize the question on scientific dimension, I think, so Pirada was mentioning very, very important elements that we share effectively in terms of next step collaboration, scientific breakthrough that we need to look at for any given disease. I think there is a dimension that we are forgetting a little bit. If you want to use AI machine learning at scale, you need to feed your model. And there is not so much that you can do internally, because once you have pushed your product in the pipe and you enter in the commercial space, never ever you will put money on research to redesign the experiment to feed your algorithm. And that's where you can play a role. Because if we were able to share database on a given disease, on a given technology, all the data you generate in the lab, cell-based assay, preclinical, clinical, if we could aggregate all this data with models that the pharma industry are investing on, we will accelerate dramatically the understanding of the disease, the mechanism of action, and down the road, launch even faster new vaccine. This is something I think we should really focus on.
Hi, thank you for that nice talk. And you touched on this a little bit. I'm Larry Madoff from the Massachusetts Department of Public Health. Can you talk about efforts to broaden the effectiveness of a flu vaccine to make it effective across hemagglutinin types or across flu strains? I know that's been a long-standing effort, but I wonder if there's any progress in that direction.
Yeah, so it's effectively a long-standing effort, and I have some people, some colleagues I didn't put on the slide, but they are here in the room to help also to answer. But basically, it's been a while that we are internally really focusing on what we call universal flu. The difficulty with that is that you have a kind of retrospective validation of this approach every single year. If you want to come with or replace a strain that WHO would potentially recommend, it will never be accepted by authorities, at least at this stage, because you are retrospectively validating your assumption, right? Now, the approach we are taking, that's what I was showing on the slide, yes, definitively, we are looking how we can select through machine learning, a little bit like Pirada was mentioning also, what is the best sequence, the best strain that can allow to have an overall broader coverage. But the idea is to propose to authorities and WHO notably an augmented version, meaning that WHO will come with 3 strains for the trivalent, the TIV vaccine for seasonal, and you come with an additional one, demonstrating, as I showed today, that it's not, I would say, at least you don't have a degradation of the overall performance of your vaccine, but on top of that, you are expanding the coverage. And for this, we do believe that in terms of regulatory pathway, it's really the future. So we are working in this direction. I don't know if you want to add something, Kathleen, on that one.
I think your point, there's multiple ways of doing that, but I think the point is looking for broader protection is certainly something of interest if we can protect patients from the variability of strains.
I'm Lillian, everyone heard me talk earlier.
Yeah.
I think often about vaccine hesitancy, and actually I think it's a lot stronger than that now. There's really this. Yesterday, I told a couple of people I was enrolling a mother in a study, and the only thing she asked me is, "This doesn't have anything to do with vaccines, right?" And that was the only question. So it's really more than that. And I was curious, a number of different questions related to this, but whether there is, from the company perspective, and curious if this is true at Pfizer, sort of like what social media campaigning, influencer positive, I don't know, the moms who go out there and don't say like, my child got a vaccine at 4 months and is ruined for life, but like the person who goes out, how do you like, are there people on the industry side who if that is that a major investment, trying to combat that in social media platforms or other educational platforms? Is that to us as physicians? Is that to teachers, educators? Like, what's the role to try to combat misinformation and just like indoctrination that has happened pretty rapidly over the last few years?
So the most exposed person around that is here, Kathleen.
Yeah, I don't think there's one silver bullet to your comment. I think, and everybody in this room probably is on the same side of this equation. So yes, we have a diverse ecosystem that we work with, and there is, like I said, multiple ways. And I think as an advocate, you own some of that in terms of clarifying misinformation. I think what we found is there's an extreme, right, of people that there's a group that's not hesitant, that they're just anti-vaccine. But the hesitant person, if you will, typically needs to talk through, right, the details and the facts. And right now you have providers that are stretched, right? They have no time to sit there and talk through that, but really arming them. So as a company, we try to arm them, especially, I could speak to more the US market, we arm them with facts so that they're able to answer the questions with facts that are concrete that they can reference, and really trying to establish that credibility. The number one reason people get flu vaccine is because of a doctor's recommendation. So again, it really goes to that provider who takes the time and goes through that. We also know that there's a concerted effort in certain ecosystems. Those of you who may know, like LJ Tan, he's like a huge advocate for flu, and they now, I'll say troll, but in a good way, troll misinformation. And you have these like American Academy of Pediatrics, or not ACFP, CDC, other organizations, AFFP, gosh, I have to retrain myself to not say ACIP, but basically come in and combat that with no, let's talk about the real facts, and shedding light on the facts, and just being a little bit more aggressive on that, because it seems so loud. And unfortunately, here, the louder voice is getting the credibility, which is not really the place to be. So as a company, we certainly are trying to put our information in the hands of those that are at the forefront, but it really comes down to making sure that we're in all those locations and having those doctors feel supported, or anybody who's intercepting the patient feeling supported with the right material. But it's really just, don't get tired. You got to keep saying it and repeat it. So hopefully that helps.
Completely agree, but I want to emphasize the role the providers have, because a lot of times, while industry is doing our part, the perception is that we have a conflict of interest. So the more people hear from independent, groups and organizations and providers, I think that also kind of generates a different perspective, right? I mean, Pfizer can come out and just say, like, vaccines are great, but like, yeah, but you make vaccines, so you're only trying to, improve your business. So I think the importance of lay people, providers, organizations, et cetera, is extremely important.
Very good point.
We'll take these two additional questions as well.
Hi, Seth Redmond from the Yale School of Public Health. A lot of these things like updating mRNA vaccines or model training rely on genomic surveillance, and outside of flu, that's kind of ad hoc and done by this hodgepodge of academia and public health labs, a lot of which are being cut back. So kind of a question for you, and Pirada, like,
can you speak a little bit closer to the mic?
Sorry. A question kind of for both of you. What is the view of Pfizer and Sanofi on maintaining this level of genomic surveillance when it's not being supported at the national level?
You want to start? Women first.
I think definitely some of the surveillance issues are kind of within the US, where there's a little bit of decrease in surveillance that we're seeing here. I think still the importance of strengthening the surveillance system, working with the WHO. I know the US, is going to not be part of the WHO, but I think there's other means like IFPMA, et cetera, where manufacturers work directly with some of these other organizations to strengthen the surveillance system. We absolutely need the surveillance to keep our defenses in place.
Yeah, same type of answer. I mean, definitively surveillance dimension is critical, and we cannot afford to downsize this aspect. So,
of Jake, and then one last question.
Hi, I'm Jake Lemieux from the MGH Division of Infectious Diseases. Really nice talk. So shifting gears a little bit, I wanted to ask you about the monoclonal antibody experience. You mentioned nirsevimab or Beyfortus, and it's really had a huge impact, as you said. And I was wondering if you could contrast the success of that to monoclonals that worked for a time and then really didn't work any longer in SARS-CoV-2. And I'd like to get your perspective. Is that something about the virus, a particularly good monoclonal? What's the prospect for monoclonals for other viruses looking forward?
I'm probably not the best position to answer deeply scientifically on it. I don't know if Josh is in position on that, but what I'm going to tell you today is that definitively what we observe, notably with experience on Beyfortus, if you design properly your antibody, you can really make the difference. And we want to contemplate at our level, and we are reflecting on continuing this type of journey for other type of disease. And we do believe it can make the difference at the end of the day. So there is a lot of effort on it, definitively. Josh, do you want to complement with something? Or?
Yeah, I mean, I can speak, specifically to the antibody you're referring to with RSV. We have a situation with RSV where it's a relatively invariant antigen, right? There's some variability in the epitope that's recognized. We obviously have that under watch at all times. Much more challenging with things like COVID, flu, where there's a much larger antigenic space, a lot more variability in the key epitope. So yeah, I think there are targets where you can be successful with an antibody. There's also opportunities to try and target very specifically the invariant epitopes with a monoclonal, but that's challenging with targets like flu and whatnot. So.
Last question.
Hi, thank you for the talk.
Closer to the mic, please. Thanks.
Okay. So I guess we will all agree that we were not prepared for the COVID-19 pandemic, right? And you talked about how we can be more ready. But when it happened, which is like quite recent, we had the technology, even though we didn't have maybe the same technology or same toolbox, we were still kind of advanced, and humanity experienced pandemic before, right? So what did we learn specifically from the COVID-19 pandemic that can accelerate our success for a future pandemic? So again, you mentioned like global collaborations kind of things, but there were collaborations. So I'm wondering what was your intake?
Yeah, I can give you some perspective, and after Kathleen, if you want to jump in. From what I saw in Europe, because I'm based in France, a little bit in the US, but What I saw in Europe is that the complexity we have when you want to prepare an organization, meaning governmental offices, private companies, etc., payers, etc. You need to have a kind of real example or live example where people can play with it. It's like a little bit when you have a fire alarm in a building. If you are not trained, it's going to be a panic, right? Here, that's what we observe in Europe currently, and even 2 or 3 years ago, people knock at the door of Sanofi saying, we want really to have a pandemic readiness plan. So we know that we have to do A, B, C, D, and it will work. And one of the elements, just to give you an example, was how to keep a facility warm. So the ask from the authorities and the government was to say, we want you to build a facility, notably the famous modulus you have seen. We're going to pay a ballroom, so it's roughly 200 or 300 million euro, and we want Sanofi to keep it warm. So keep it warm, is it to put a heater in the room and keep it warm, or is it to have people really working in the facility every day? If you want to be ready, you need to have everybody working on the topic almost every day. And when you take a facility like the one I was talking about, it means that you have to pay 35 people full-time to do nothing. If you want to have a warm facility, it costs you a huge amount of money that the government are not ready to put on the table at all. When you tell them, fine, I can build it, I will put 35 people to stay trained on the technology, nobody will pay that. And that's why the beauty of having technologies like mRNA, eggs, that's another story, because Sanofi has huge capacity for decade on eggs, so we can switch rapidly, but it's very specific, right? But mRNA techno allow you, as you're going to apply it on different type of target, that hopefully going to go to commercial space, people are trained and people are ready. And all of this need to be activated at every single step of the value chain, from the governmental policy. So we are talking about surveillance, etc., etc. Have all the tool to just push the button the day it happened, definitively. And after, there is a notion of communication also between the different actors, stakeholders, that sometimes is at that time was a bit broken. Kathleen, do you want to add something?
You covered the aspects I'm sure Pfizer could say as well in terms of the challenge. Okay, so I think the only other aspect about keeping the facility warm is that if you're talking about something like 1918 and you have over 8 billion people to immunize with 2 doses, to even a warm facility, you're not instantaneous to get around the world. So, and I think the further away we get from pandemics, right? You had 2009, much more mild, different population, but the further away you get from it, people don't have memory. Think about how much you spend on insurance, right? So you're like, do I really need that insurance as time passes? So I think, yes, people learn, but the further away we get, we relax. And if you're trying to turn on a dime and you're going from 0 to 100, and that's very, very difficult to do. So, but again, I think the partnerships and things that we're doing will help put us in the best position possible for the next pandemic.
All right, that was a wonderful talk, wonderful discussion afterwards as well. That was terrific. So we're going to move on to our first plenary talk of the second session. Is going to be a team. So it's going to be Louise, Hansen, and Karan Malhotra. So Louise holds a PhD in bioengineering from UCSF and UC Berkeley. She works on developing new diagnostic workflows for infectious disease. Dr. Karan Malhotra earned his PhD in analytical chemistry from the University of Toronto and developed accessible, ultrasensitive diagnostic technologies for point-of-need testing. Both of them are postdoctoral fellows in the Walt Lab at the Wyss Institute. So welcome, Louise and Karan.
Awesome. Thank you. Okay. My name is Louise, and this is my colleague, Karan. We're grateful for the opportunity to share our work on developing an ultrasensitive multi-analyte detection assay for predicting disease trajectory. I want to start out by briefly revisiting the early days of COVID-19. Across the world, patient demand surged far beyond what the hospitals were capable of handling. We ran out of beds, ventilators, critical drugs, and clinical staff really bore the burden of this and were rapidly overextended, which forced triage decision-making, and in the worst of cases, crisis standard of care. COVID-19 waves created this severe mismatch between what we were able to handle in the hospitals and the patient demand. Hospitals became the front line for pandemic preparedness, but lacked the quantitative tools to guide resource allocation. So with that in mind, our goal for this project is to enable data-driven resource management for future pandemics. We want to empower clinicians to identify which patients are at risk of progressing to severe disease. So ultimately, you can shift care from a reactive method into a more proactive method during periods of constrained capacity, such as during a pandemic. Today, we're presenting a diagnostic solution that would allow hospitals to do just that. It would allow clinicians to predict the patient outcome and allocate resources accordingly. In a more effective way for future pandemics. Our solution is designed for accessibility, adaptability, and performance by integrating 4 key design features. Our analytical assay requires small sample volumes. It's run on widely accessible equipment, such as flow cytometer, so that it can integrate easily into existing infrastructure in hospitals. It can be implemented as a shelf-stable reagent kit and can measure low protein abundances. We're combining all of these into the ultrasensitive field-deployable biomarker assay that we are presenting today, which will enable a prognostic model. That model can help doctors predict disease severity for an individual for pandemic preparedness. In other words, we really want to give clinicians an early indication of who might need additional intervention at that first testing opportunity. They're coming in, getting tested for COVID. Can we predict the likelihood of severe disease progression so that hospital resources can be used strategically?
Now I will describe the protein detection platform that we've developed with support from MassCPR, which we call EF Mosaic. The EF here stands for enzyme-free. And MOSAIC stands for molecular on-bead signal amplification for individual counting. EF Mosaic is an ultrasensitive detection technology. It measures, it has the ability for single molecule detection. So we're measuring the lowest concentrations of proteins possible. Using this approach, we can achieve detection limits that are approximately 1,000 times lower than bulk ELISA methods. Our approach is to use these dye-encoded magnetic beads that are conjugated with antibodies. These beads are then incubated with patient samples to capture individual protein molecules. As you can see from this figure, there are beads that have one protein molecule and beads that have no protein molecules. What we do next is we use biotinylated detector antibodies and streptavidin conjugated signal amplifiers to label the single protein molecules. We can then take this bulk of beads and process it via flow cytometry to get a readout. In our workflow, we don't use any enzymes. Enzymes add expense. They can lose performance over time. They require a cold chain for transport and storage. And eliminating all those things would greatly improve the accessibility of this technology to those in need. Once we label the single molecules, we measure them with the flow cytometer. Unlike specialized imaging equipment, flow cytometers are widely accessible. There are initiatives and organizations like Flow Africa that are actively working to enable laboratories in low and middle income countries to use flow cytometry. And we believe that having a flow-based readout would increase the accessibility of this technology. Samples can be processed in a few minutes. We can get high throughput readout for multiple patients. And we can greatly improve the translation of the tests to the patients. I will now pass it to Louise, who will describe a bit more about the type of data we get from these types of measurements?
Yes, so to implement EF Mosaic for pandemic preparedness, we started by developing 7 assays for different antigens on the EF Mosaic platform, proving our worth against the COVID-19 pandemic as our proof of concept. Here we're showing the calibration curve for representative cytokine A as measured with EF Mosaic. You'll see concentration along the x-axis in the units of picograms per mil, and the average fluorescence per bead across the y-axis here. The vertical line marks the limit of detection of conventional ELISA, and you can see we dip below that for this assay, and we do for our other assays as well. EF Mosaic achieves substantially lower limits of detection across all of our targets, extending into the sub-picogram range. The sensitivity here is the foundation for meaningful prognostic modeling coming up, and the mixture of measuring cytokines and viral antigens will give us a chance to kind of proves our ability to measure a range of different biomolecules with different biological function. So to quantify both pathogen presence and host immune response, which is what we want to do to predict severity, we combine these 7 antigen assays into 2 complementary panels. One is what we're calling the viral antigen panel. This is what gives us indication of a viral infection, specificity, and the pandemic relevance of these assays. To complement that, we have the host cytokine panel, which indicates immune activation and essentially tells us the severity of the response within the person. We have 3 viral antigen markers in the viral antigen panel, 4 host cytokine biomarkers in the host cytokine panel. And together, these modules provide the what of the infection, such as the pathogen, and the how bad of the body's response. We're using this modular design for rapid assay adaptations for future pathogens. We envision that if a new threat comes up for pandemic potential, you can drop out the viral antigen panel we built for COVID-19, drop that out and plug in a different panel for a different virus of interest. So in order to then convert from these 7 singleplex assays into these 2 panels, the cytokine panel and the viral antigen panels, we need to generate multiplex assays. In EF Mosaics, we can multiplex by using different color-coded beads, and where each bead encodes for a different biomarker. So in this case, we can do simultaneous detection in a single reaction of the 4 host cytokines by having 4 different colored beads where each of them corresponds to one antigen or to one cytokine, and we can measure them simultaneously using kind of an unadjusted EF Mosaic workflow. And we see no hit in terms of performance on these assays when we move into multiplexing format, when we preserve sensitivity and quantitativeness. On the flip side of that, for the antigen, we can combine 3 different colored beads to measure 3 different antigens, do that in a single pot reaction, and again, we see analytical performance that matches their singleplex version of these assays. The benefit of keeping the 7 biomarkers as these 2 separate panels is, as we mentioned, an ability to switch out a segment of them or one of these panels for a different virus, and it allows us to do sequential pull down to optimize what information we can get out of a very limited sample.
So sequential pull downs are when you do 2 multiplex measurements in series. This allows us to avoid the high cross-reactivity of a single 7-plex panel. And it also allows us to maintain the modular framework that we use to design this assay. In the first panel, which is the cytokine panel, we take patient samples and we incubate our beads for a period of time. We then transfer the sample to a fresh file. And because our beads are paramagnetic, we can actually retain them and rehydrate them in buffer for analysis by EF Mosaic. Separately, we take the transferred sample and we incubate our second panel of beads, which is the COVID panel in this case, and we process that using the same workflow as a regular multiplex assay. So what the data shows us here is we took 2 different assays from both of the different panels, and we ran them in the pull-down format and the multiplex format. As a consequence of the pull-down workflow, we actually see no loss in performance when compared to the multiplex assay, and we can then retain our modular framework and use it for the next pathogen. So having investigated the multiplexing workflow, we then looked at the ability to deploy this technology for measuring it in a field setting. We looked at, in order to make EF-Mosaic widely available, we looked at the stability of the streptavidin signal amplifier reagent, which is replacing our enzymes in these assays. We looked at stability at room temperature, because eliminating cold storage and cold transport can make this rapidly deployable. And we also looked at the stability of our reagent at extreme temperatures of 50 degrees Celsius, which it was stable at for 48 hours. As you can see, we had no loss in performance in either of these conditions, which suggests that we're on the cusp of being able to deploy this technology for the next pandemic. Our next steps are focused on using the data that we have generated from EF-Mosaic as inputs for a prognostic model. Our goal is to enable the stratification of patients by their disease severity, which we're trying to do in collaboration with the MGB Biobank. Using the MGB Biobank, we have access to samples from patients that have anywhere from a mild reaction to COVID-19 to a severe reaction to COVID-19. We can then take their reaction and their health trajectories, and we can use that as an input along with the results from our EF-Mosaic assays to develop the prognostic model. This is our next step. We're really excited to do these measurements. We're getting the patient samples this Friday, and we hope to share them with you very shortly. We would like to thank our colleagues in the Walt Lab and our PI, Dr. Walt, for their support with this project. We would also like to thank the MGB Biobank for helping us with the patient samples, and the MassCPR organizers for allowing us the opportunity to share our work. Thank you.
Wonderful talk, guys. I'm just going to start with a question. Thinking about pandemic preparedness, hypothetical pathogen emerges. What's the timeline for, say, pathogen identification to potentially having something you could roll out to for clinical use?
Yeah, I think I'll start with that. So with our technology, it's an antibody-based ELISA-type system. So we would need access to antibodies, and we would need to identify the specific viral antigens that can give us the specificity we need to detect new pathogen versus the previous pathogens or strains. So once we have that, we can actually use that as a drop-in module for our overall system, and we can then just get that specificity and then use the cytokine panel to figure out the severity of the patients. So the timeline really is based on when we can have accessible reagents, particularly the antibodies and the protein standards.
Yeah, and maybe to jump off of that, one thing we're hoping can give us some flexibility is that you don't have to pick, if the new pandemic virus of a type appears, you don't have to pick a single antigen that you think can be like your one and only, because we can multiplex pretty readily in this system. And we did 3-plex and a 4-plex in this demonstration, but we've also on similar systems shown 8-plex and 12-plex. You don't have to pick your one and only. We could take a more of a high or more of a kind of spreading out your odds by going in and taking, the 4 best antibody pairs that are coming out instead of waiting for one fantastic one.
Mm-hmm. Yeah. And because we have like the sequential pull down framework, we could even look at, say, pathogen A as a module, pathogen B as a module, and then the patient response. So there's a lot of flexibility based on the workflow.
Yeah. Thank you for your question.
Hi. I realized I forgot to introduce myself the last time I asked a question. I'm Ellen. I'm from the Needle and BMC. And I'm curious, given that different viral diseases have different biomarkers for disease severity, for deployment of this in a new pandemic setting, do you have thoughts on how best to accelerate the identification of biomarkers to add into that panel?
Yeah, that's a good question. I think in the development phase, we would have to look at, panels and see what really works with most patients, and then see if we can use that as a drop-in. I think definitely having that flexibility of, say, picking panel A and then panel B really allows us to design the assay first and then focus on implementation. So it would require some type of a pilot study.
Well, thank you. You talked about using this as really a clinical tool, but for research we also need cheap, stable reagents that are flexible. So do you see the application for even making this technology available for using in the lab and for being able to mix and match a little bit more your antigen or of interest?
Yeah, for sure. I think Karan and I are really motivated by the idea of enabling other scientists to pursue tougher scientific questions or to get the kind of answers that they want. We would love to be able to hand this off to a lab and help them establish what kind of biomarkers they're interested in measuring for their specific focus. It's not that hard to do, and we're really optimistic that that could be a fantastic utilization for this kind of technology for sure.
Yeah,
and that could kind of help us to the previous question, right? If we get more people looking at these viruses, more people looking at the host responses, things like that, maybe that will make it easier for us to then pick the right panel for the next one. Yeah.
Great, thank you guys so much.
Awesome, thank you so much.
All right, we move on to our next speaker. So it's Adam Sychla. He's a postdoctoral researcher in the Ruskin Lab at Harvard Medical School. He completed his PhD at the University of Minnesota in 2023. Adam.
All right. Well, thank you everyone for inviting me here today to speak at MassCPR. I really appreciate the invitation and the funding over the past year to work on prediction tools for zoonotic spillover in Picornaviridae. And so this project was really inspired by the fact that viruses create planet-scale effects. And these effects are actually just encoded in a very minimal amount of information. I took the liberty of encoding the enterovirus A71 genome as a PNG, and that's the entirety of it, and that stores about 3 times smaller than an empty Word document. So there's not a lot of information that fully determines whether a virus will spill over, whether it'll become a pandemic or cause an outbreak. And so I wanted to lead a project where we went from this minimal amount of information to actual predictive power. And doing that for all viruses is maybe a little bit too ambitious of a goal. So I narrowed down to the family of Picornaviridae. And I chose this family for a number of reasons, one of which is, for starters, they actually express in a wide range of tissues, and sometimes they replicate in a wide range of tissues, and sometimes with rather high specificity. They are single-stranded, positive stranded RNA viruses, and importantly, they produce their protein using these complex structures called internal ribosome entry sites, or IRESs. And these IRESs do determine whether a particular virus could replicate in a given cell type, for example, human cells. And so in this way, we can actually we have a measurable readout, which is iris activity, that at least has partial predictive power of whether there's a given virus has even the capability of spilling over. And so to that end, we wanted to measure the activity of many irises in a wide range of tissues and cell types. And we used a reporter system based on circular RNAs. And in doing so, we have a platform where we have a barcode, the IRES, and then the luciferase reporter. And this splices into a circular RNA construct. And because these each have a unique IRES and barcode, we can multiplex this. We can deliver 100 IRESs all at once. Indeed, we did that with 96 because gene synthesis formats are in 96 format. But we designed we picked 96 IRESs, about half of them from human cells, or that are native from human-hosted viruses, about half from a range of different animals across all 6 IRES types. And in a single experiment, we were able to measure the activity of all 96 of these IRESs here in 293 T cells. And we get quite a bit of range. We have these 2 viruses down here, irises at the bottom, that have essentially no activity inside our system, and then up to the Caprine Cobo viruses up here at the top. And we were able to see that there's quite a range right away for industry applications. EMCV, which is the IRES that you usually see when you order something from Adgene and says IRES, it's probably this one. It is actually about 2-fold weaker than some of our top performers. Now, taking a look, we have this is data for one cell type, and I can collapse that into one row here. But then we have the same data across multiple cell types, and we have different activity for each IRES. And what stands out right away is that we have these different clades and clusters of expression profiles. So for example, this blue cluster over here has higher translation efficiencies in mouse neurons, versus this purple clade has a lower translational efficiency as opposed to in other cell types. And then I'll point out this yellow cluster that has essentially no activity in U2OS cells, which are osteosarcomas, but then it has a level of expression in a few other cell types. And when we do the stats, most of IRESs actually have significant tropism, i.e. preference for a certain cell type. Now the scale of this can range. I use Gini index here as a scale. But since Gini index is also used to measure economic disparity, the US is floating around 0.3. So that will be kind of the scale of the effect here. Now, like I said at the beginning, the goal here is how can we go from a small amount of information to a functional predictive readout. And to that end, we decided to approach this using a IRES-directed large language model. Essentially, we took a large language model that had been previously trained on 36 million non-coding RNAs and gave it just sequence information from 500,000 additional IRESs. So for all intents and purposes, I went to NCBI, downloaded 500,000 sequences, partially from NCBI, partially from blasting further, and it was only trained on this information. And what we saw from our model is that we can actually pull out not only sequence information, so it can detect and predict sequence motifs that are conserved and or important, but it can also actually report back on structural information about these IRESs. It can predict what structure these IRESs are folding into. And the way we do this latter part is building off of a recently reported approach by De Silva et al. Called dependency mapping. And in dependency mapping, basically what we do is we take every base inside the IRES, mutate it, and ask the large language model to predict the other base. And the reason this works at all is that we can use a similar language kind of metaphor and say I have the sentence, I have a glass of, you might fill that in with water. However, if I mutate the word glass into bowl, you would say, well, I need a bowl of soup. So we would say that these two words or these two bases have high dependency on each other, which would be predicting of a base pair forming there. On the other hand, if I mutate the word need into want, you would say I want a glass of still water, and so there wouldn't really be a dependency between these two words and or these two bases. And why that's useful at all is that when there's these dependencies, we can actually predict structural information from it. These anti-diagonals are indicative of a stem forming in the RNA structure. And here I'm showing EMCV, like I mentioned earlier, it's kind of the default IRES. And we can take this well-studied structure that's well-known, has been is validated by DMS data and many other forms of experimentation. And all of the structures that are predicted from previous experimental data are recapitulated here in this portion of the IRES just from our model alone. Now this is a EMCV, which I mentioned again is really common. There's many, many examples. So of course our training set has many, many examples that are similar to this. It was learning on this data. But I would like to point out a different edge case where we worked on this virus called Galavirus, infects chickens. And the sequence that we were looking at is actually very divergent. So in our entire training set, we only had 4 examples that aligned at all to this virus. And within those 4 samples, they had 100% match within this region. So it was really learning on 4 examples that looked exactly the same. And it was able to predict this structure over here, which is the one that has been previously reported from other data, and also matches our DMS data, which is a measure of how likely a base pair is, or how likely a base is to be paired or not. So you can see the low reactivity bases have low DMS signal, and those are the ones that form in the stem, versus the high reactivity form in the loops here. And importantly, this GNRA motif that is known to really form around this sort of structure, it is right where we would expect it to be. On the other hand, we've also run a covariation analysis, which was unable in this pretty edge case. Admittedly, like I said, this is a highly divergent virus. There's not a lot of data for it to be working with, but it predicted this other form here that doesn't quite match. And so it shows that our model here is actually learning larger patterns about how IRESs function, not only on the structural area side, but also on the sequence side. So moving from here, we're really excited to adjust this pipeline to be a pipeline for rapid secondary structure prediction of really any RNA class that we can just download sequences and train on. The training is quite fast, and we can then put out answers. Importantly, matching this large language model prediction of structure and sequence motifs, we can actually match that to the activity that we had measured in a range of cell types earlier. And this gives a readout for spillover risk by identifying which IRESs even have the potential to be translated in human cells. There's some potential for long-scale interactions. And again, this can be used to design IRESs that have tissue specificity and then matching to our activity data, and by extension, be a way that we can control RNA therapeutics so that they express in the tissues we would like them to express. With that, I of course want to thank my lab, thanks to Pierre and Grant and Jacob, who are some of the people that were working with me on this project and had done a lot of work here to make all of this data happen. So.
Going to encourage people to ask questions, and just to kick it off, lots of potential applications of the model. In the context of pandemic preparedness, spillover risk, where exactly do you think this would maybe fit in with surveillance programs? Like, how do you see it fitting in there?
So one of the great things is that once we have the model trained, we can actually run it in less than 2 minutes. And so if you were to sequence a new IRES out there in the wild, you could enter it as long as you have an internet connection. You can enter it and it'll run immediately. give a few minutes and it would be able to spit out at least some amount, or at least the structure sequence information. And as we're building up the capabilities for prediction of activity, that would be in the order of minutes.
Really interesting work. The picornaviruses have such diverse clinical manifestations, like, poliovirus sometimes causes neurological manifestations, other times asymptomatic, or just causes a mild febrile syndrome, and really enteroviruses, rhinoviruses, just the incredible diversity of hand, foot, mouth disease, and enterovirus D68, for example, that is neurotropic. So my question is, do you think any of the tissue-specific activity that you're seeing for these IRESs could explain some of the rich clinical diversity and manifestations of viruses that are part of this family?
There's already precedent for this, at least polio. There's a single point mutation in the IRES that determines whether it's neurotropic or not. And so at least at that level, that is part of the determinant of how severe you could potentially get polio. And so to that end, yes, I think there is predictive capability. There's other examples throughout literature of different IRESs that only express in certain cell types. And so obviously would have an impact on what tissues they replicate and what phenotypes you get.
Hi, this is a really cool concept. Outside of the context of irises, and I apologize ahead of time if this is a naive question. I'm not well versed in LLM training, but could you describe a bit about what sort of scope of training RNA data sets you might need to predict other types of RNA structures besides irises in viral genomes, for example, particularly those in viruses where the genome is most of the time is encapsulated, for example?
Yeah, so from the level of how much training we need, we're working right now to see how much. We admittedly went hard with going with 500,000. We know that that's not the absolute we need, and it might matter a little bit whether we're looking at an entire class, and a very diverse class here, or if we care about a more limited class of RNAs. In terms of in the context of being encapsulated, there still would be these covariations, because while it does predict structure, it's not the only thing that covariation predicts. It can predict codon usage, as well as other features about the RNA, or even what structures are there in the mRNAs that are produced during replication. And so there is still useful information. It would be extracted a little bit differently further down, but you definitely can learn that from training on those kind of.
That's really cool. Thank you.
Hi, have you tried using the model to predict RNA structures in 5' TRs and 3' TRs of messenger RNAs?
We have not yet, but yes, that is completely on our to-do list to do next. The base model that we use here, there are base large language model. There we go. There are base large. We use the Renalma, which was a base not trained on 5' UTRs or 3' UTRs. There was mostly like non-coding RNAs like tRNAs, a few tmRNAs, telomerase, but there are models that have been large language models that have been trained on 5' UTRs. So this kind of pipeline could potentially work off of those models, or we could fine tune using a set of 5' UTRs to work on that in that context from here. But we would have to do that later.
Okay, thank you.
All right, thank you guys so much. Thanks, Adam.
Thank you.
All right, then just our last presenter for the session. So this is Jared Herrera. Jared is a PhD candidate in the Mollberger Lab at Boston University's Needle. His work focuses on virus-host interactions and developing reverse genetic tools to study novel hepnipoviruses.
Thank you for the introduction. I'm very grateful to be here and provide the opportunity to share my work that I've been working on, titled Hepnipoviruses: An Expanding Threat. The first thing I'd like to do is introduce okay, it is there, is introduced Nipah virus. So this was identified in 1998 in an outbreak in Malaysia and has remained endemic to the region with near annual outbreaks of human disease. The proposed animal reservoir for this virus is Tropus species bats, or commonly known as flying fox, and some basic transmission. There is potential for transmission from bat to human, but we've commonly seen, or we have seen, transmission from bat to an intermediate host species to human. And there's also evidence for environmental factors such as consumption of contaminated date palm sap. So this pathogen is a WHO priority pathogen. So it's kind of, why is this such a concern? Why do we care about studying this? And this is a very lethal virus. There's a 40 to 75% case fatality rate. And during infections, the symptoms will start as the typical flu-like symptoms, but can progress to severe respiratory distress. And in some cases, neurological symptoms will present themselves, which can be followed with coma and death from that path. Additionally, this virus is known to have documented cases of person-to-person transmission, and the use of a highly conserved host entry receptor, the FRNB2 and B3, means there are many different routes for zoonotic transmission, as this is highly conserved across many species. And given this information, this is a BSL-4 virus. Work on this pathogen is restricted to maximum containment. Now, Nipah is not alone in its genus. In 2012, there were 4 known viruses. So Nipah, Hendra, the other lethal virus in this genus, along with Cedar, proposed non-pathogenic, and Ghana, which was largely unknown. All of them shared the same proposed animal reservoir of a bat. Now, if we skip to today, we see this tree has greatly expanded. There are a lot more identified hantaviruses. And we also see expansion of the proposed host reservoir. While initially it was bat, now we see this kind of a split between bat and shrew. Something I'd like to highlight is Camp Hill virus, which was published on earlier this year. This is the first North American hantavirus that's been identified. So with this one, it truly shows that this is a global genus of viruses. These are present everywhere. Now, the 2 viruses I'd like to focus on today are Ghana and Lanya. So to introduce these 2, a Ghana virus identified in 2008. It was the first hantavirus found in mainland Africa. Also, bat-borne, it is not known to cause disease in humans, but it is proposed to use one of the same host cell entry receptors. Additionally, sera from local people has been shown to have the capacity to neutralize Ghana and Nipah virus particles. That doesn't mean there's infection, but there is something in the region that is generating a response by the local people. Lanya virus, this is a much more recent discovery, identified in 2021. This one is known to cause disease in humans. There have been 35 documented cases of severe respiratory distress, but no known fatalities. Unlike Ghana and Nipah, this one uses the proposed reservoir is the shrew. And structural analysis of the glycoprotein shows that it likely uses a different host cell entry receptor. So one of the biggest challenges with investigating these novel viruses is we typically don't have isolated infectious virus. A lot of these have been identified through environmental sampling, where they've sequenced RNA that they've collected. And so there's no virus to work with. We just have sequences. And often we only have a few sequences to work with for each of these. Compared to Hendra and Nipah, which are extensively studied, Hendra has about 18 published full-length genomes. Ghana has 1. So there's a big disparity in available information. And many of these novel sequences are missing elements. They're incomplete, and these crucial genome ends are often missing. Now, this is not like the end of the story. We're able to overcome these issues using reverse genetic tools such as the mini genome assay and recombinant virus. Just a quick rundown of the Henipavirus genome. There are 3 proteins of interest for this, the nucleoprotein, phosphoprotein, and the polymerase. These 3 combined make up the replication complex for these viruses. And this will be important in a moment. For actually making our tools, the mini genome and the recombinant virus, we take these genome ends, so the 3' called the leader and the 5' called the trailer. These regions contain crucial promoter elements for the viral polymerase. So without these, we won't get transcription and replication of the viral genome. So for the mini genome, we take the genome ends and we will sandwich a reporter gene between them. And for the recombinant virus, a bit more straightforward, we'll take the viral genomic sequence, put it into a plasmid, and in some instances include a reporter gene that is fluorescent for a measurable activity. Now, in use, the mini genome, we take our reporter gene with the genome ends on the outside, we transfect that in with the components for the replication complex into cells, and we'll get production of our reporter genes. Conversely, with the rescue system for the recombinant virus, it's performed in much the same way, where you take your viral genomic plasmid, transfect that with the replication complex, and it should produce infectious virus. A key distinction is the mini genome is BSL-2, as no infectious virus is produced, and this is a great tool for high throughput analysis for testing viral inhibitors. You can really screen a lot of different conditions in one go with this system. The rescue, since you're generating virus, is limited to the BSL-4. Now, so this is kind of the rundown for the tools, but in practice, with the longest system, I have made a very functional and productive mini genome system. We use a dual reporter mini genome, so we have a fluorescent Z-screen reporter along with a quantifiable nanoluciferase. And we can see that we get great production of our fluorescent protein and a good measurable amount of our nanoluciferase. So given this is functional, this tells us that both the genome ends and the replication complex are functional, which bodes well for generating a full-length infectious virus from the sequence. For Longya, this is still ongoing. But to move to Ghana, we see that it's one of those viruses that is incomplete. We are missing about 28 nucleotides from the 3' genomic leader. But we're able to kind of get past this. Looking at the closest relatives to Ghana, Cedar, Hendra, and Nipa, it's important to note that these 3' genomic ends are very conserved between these species or between these viruses. And so they're all roughly the same length of 52 nucleotides, and these promoter elements are also very conserved between them. So the method we used here is we complemented the missing Ghana sequence using either Cedar or Hendra. So this missing section, we took this sequence and incorporated it instead. So using that, when it comes to the mini genome, we see that the Cedar complemented mini genome really doesn't work. We don't see activity of our fluorescent reporter or the nanoluciferase, but the Hendra complemented mini genome is active. And we see that the same thing is recapitulated where we get quantifiable amounts of our nanoluciferase. So again, like Longya, this means that our genome end, the replication complex is functional, and this complemented genome end is recognized by this Ghana viral replication complex. So moving forward from here to generate a full-length infectious virus, our schematic is something like this, where we have our complemented genome end along with in this case, fluorescent reporter that we've incorporated. And much like the mini genome, we transfect cells incorporating our replication complex components. I was able to generate infectious virus of this novel Henipavirus. So this is just tracking the same region over time. We do see that it is spreading. And while not present in these images, I did see novel infection sites in the flask appearing, which suggests that this virus is budding and is able to cause new infection sites, and it is not limited to cell-to-cell spread, indicating that this is the full virus or functional virus. Now, I've made this virus, but The main thing is we don't know anything about it. We don't know how it functions. So the first thing I wanted to test was cell tropism. So I tested a variety of cell types, starting with hamster, shrew, bat, and monkey or primate cells. And we see that this virus, it grows very well in these hamster cell lines. We see a growth in the shrew cells, which I find interesting, as this is the other common proposed hantavirus reservoir. So I thought that was kind of neat to see that. What surprised me is these bat cells, which are eidolons. This is from the proposed host reservoir. It didn't grow very well in the cell lines. That caught me a little off guard. I didn't expect that. But we do see little bits of infection in primate cells. But we're interested in human disease. So I did infect a human cell line, HUVEC turrets in this case, and we see that there is good infection of these HUVECs. These are endothelial cells, and this is important because endothelial cells are a prime target for Nipah virus infection. But this is an immortalized cell line. So there's potential differences between how this will behave compared to primary cells. And so we do have preliminary results of infection in a primary-like cells using human iPSC-derived endothelial cells provided by the Cotton Lab at the BU's CREM. I did compare gonorrhea growth to Nipah, and this is very preliminary. We need there's further optimization of this assay to be done, but the main takeaways is Nipah really infects these cell line or the cell type, while Ghana is capable of infection, but it seems to be much slower for some reason than Nipah. Now, there are many reasons for why this could be. Now, one of them, Ghana has the wrong leader sequence. Maybe that's inhibiting viral replication. But another thing that I think could be in part responsible is hantavirus infection, specifically Nipah, causes a really strong interferon response in these endothelial cells. And while we know in Nipah infection, this is not protective, Nipah is able to get past this innate immune response, likely because its own host proteins are very efficient at antagonizing the host innate immune system. But it is possible that Ghana virus does not have the same capacity to antagonize the host innate immune system. I've seen in many different in other cell types I've tested infection, there will be initial infection where you can see that, it does get into the cells, but it doesn't spread, and eventually it will kind of die off, and it seems the infection is cleared in vitro. So for future directions, I am still looking at generating a recombinant longivirus. While we do have a functional infectious Ghana heterocomplemented virus, it does have a reporter gene in it, and so we are interested in removing that reporter gene and generating a wild-type virus, as inclusion of these reporter genes can attenuate viral infection. Additionally, I want to test Ghana virus infections in a wider variety of these human iPSC-derived cell types to Nipah virus infection, specifically looking at relevant to infection cell or tissue types, where lung tissue, so alveolar type 1, type 2 cells are of interest, and in nervous tissue, since we know Nipah has the capacity for neurological effects, testing in eye neurons and astrocytes. So to summarize, new hantaviruses are being identified at a pretty quick rate, and some of these are capable of pathogenic potential. Longya has documented cases of human disease. Now, these reverse genetic systems are efficient tools for studying and generating these viruses with no known isolates. Ghana virus has the capacity to infect human cells, but seems to be reduced compared to Nipah. And the Longya virus mini-genome is highly productive and could be utilized for drug screens targeting the replication complex. So if there's any inhibitors you'd like to test, we are very open to collaboration on that aspect. And with that, I'd like to thank all the people who supported me in this work and the funding sources of the Gates Foundation and MassCPR. Thank you.
Great job, Jared. Encourage questions. Just want to start. Great to see how you rescued Ghana. We've seen that kind of Hendrick, 3' UTR region. Just curious what you think about the caveats of incorporating that sequence. Is there potentially differences in tropism? Is there any limitation potentially? Do you really need the kind of native content sequence to?
So I do think that having the Hendra system or the Hendra complement, there is a reduced capacity for the virus for infection. Other work shown by the Lee lab at Mount Sinai have shown that the Ghana virus replication complex seems pretty discriminatory in what it'll accept. So I think getting a wild type sequence in place, I think could improve infection.
Have you considered doing library screens for that short sequence to figure out what mini genome works?
You mean like make like random, like a...
Yeah, mutations across that region. I mean, you can't do full, but you could do partial.
Yeah, I've considered that for some of the other work that I've done. It's a bit, for the methods that we use, it's kind of difficult to make enough of them that are like to test. Because at least for what the methods I have used, making multiple of these mini genomes could be very time consuming and laborious. I am interested in serially passaging the virus that with the Hendra complement to see if over time do we get changes in that leader region, and then incorporating those changes in a new virus and seeing if that does improve the infection capacity.
Great. Jared, thank you so much. I just want to first start by thanking all the speakers for a really terrific symposium. So maybe we can give all of them one more round of applause. Just for There's some wonderful discussion as well. I'm now going to hand it over to Nadine. So I'm just going to introduce Nadine Fornells-Martins. She is the Executive Director for Research Initiatives and MassCPR at HMS. She holds a PhD in bioengineering from the Université Catholique de Louvain, and I'll pass it over to Nadine.
We're doing really well with time today, and I will also be brief, so this is a perfect ending, I'd like to believe. So MassCPR symposia are typically closed by George Daly, Harvard University Dean at the Faculty of Medicine and Chair of MassCPR. Unfortunately, he couldn't be with us here today, but I know that he would have begun with thanking each of you for participating in this year's symposium. It has been an inspiring day, one that reflects the depth, diversity, and momentum of our infectious disease and immunity community. Each session has highlighted the power of bringing different perspectives together to confront complex challenges. What we heard today reinforces a truth that shaped the opening of our program. Progress depends on partnership, whether it's decoding the mechanisms of host pathogen interactions, translating discoveries into vaccines or therapeutics, or building new platforms for diagnostics and surveillance. None of this work happens in isolation. Just imagine trying to make a discovery and carry it all the way to the finish line alone. We know too well the challenges that come with this line of work. The need to balance discovery with urgency, tackling technical hurdles, dealing with limited funding, managing career uncertainties. But what if collaboration could turn these obstacles into opportunities as well? The pandemic, also known as a major health crisis of our lifetime, showed us that we do better together. Technologies born in academic and national labs were quickly refined and moved to mass production. So why not carry this model forward and continue building the future through shared effort and innovation? As we close, I hope you carry forward the energy and ideas sparked today. Stay connected with one another. Explore opportunities to co-create, not just coordinate. Keep asking questions and invite partners who can help answer them in new ways. Your findings can only make a difference when they move beyond the bench and into the world. There is still much work ahead to ensure we are never caught off guard again. Thank you to Bruce Walker for hosting MassCPR at the Ragon Institute today, and Gaurav Gaya for being a wonderful moderator. Thank you to our speakers and trainees for their excellent presentations, to our supporters for making this symposium possible, and to the many hands behind the scenes whose work brings this event to life each year. We wouldn't have an annual symposium without Lauren Hecht. Thank you to our in-person and virtual audiences, and thank you to all of you, researchers, clinicians, industry partners, students, and others for the work you're doing every day to push the field forward. You are a MassCPR. I also want to recognize the visionary and determined leadership of George Daly, David Golan, Jeremy Lubin, Arlene Sharp, and Bruce Walker, who drive this mission with knowledge and resolve. It is my pleasure to officially close MassCPR's 4th annual symposium, and I invite everyone to join us for reception in the lobby. Thank you again for being part of this remarkable day.